to start with this session. I'm Nicholas Wilber. I will be the chair of this session. As members of the jury, we have Karim, we have Mare, and we have Gonzalo. So, as you already know, these are 15 minutes presentations. When you are around 12 minutes, I will raise my hand like this. So you know that you have to think in finishing. And if you go to 15, I will get up and you have to finish for sure. Okay? and that's it. We are going to start now. And the first presentation is going to be given by Mehak Sangani. I pronounce it well, I think, more or less. And so the floor is yours.
Okay. Good morning, everyone. Thank you for coming to listen to my colleagues' presentations. My name is Mehak Sangani, and I will be talking to you about my master's thesis, where I was assessing the distribution and relative abundance of groupers and snappers using baited remote underwater video survey systems, or BRAVs in short. Oh, I don't think I did it right.
You can take it, yeah.
Oh.
Wow, that would keep on.
Can you just, do you want to just do it? Oh.
Swipe. Yeah.
Oh, okay. So to introduce my species, I was looking at groupers and snappers, and they are prominently large predatory fish that exist primarily in the tropical and subtropical regions worldwide. They are really important because of their high economic value fisheries, which means they also support a multitude of livelihoods. They are ecologically significant because as predators, they have top-down controls of the ecosystems they live in. They're also interesting because they have very unique, very interesting life history traits, which are characterized by slow growth rates, late maturation, and seasonal spawning aggregations. And in the case of the groupers, they also show protogynous hermaphroditism, which means that they mature first as females and then transition later into males. So because of these reasons, the groupers and snappers tend to be good indicators of ecosystem functioning, which means that if an ecosystem is doing well, we would expect to have high levels of groupers and snappers in them. No, but I want the animation to go. Oh, okay. So despite their importance, we see, and their significance, we see that they have declining populations, mainly due to anthropogenic activities such as overfishing and overexploitation, habitat destruction, and of course, anthropogenically induced climate change. Over here we see a picture of a grouper, which is the Cephalopolis argus, which is very common in the reef that we were examining. So like I said, these species are incredibly important, but there is a lot of lack of data concerning them. So it's the motivation behind this research was to address data gaps to ensure effective conservation and management strategies, which are very urgently needed to ensure the sustainability of these species. So for my study, I have two main questions. I'm sorry, I'm really bad at this. The first one being, what is the relative abundance of groupers and snappers within the Kayankarmi Marine Sanctuary? And the second one being, what are the habitat associations of the same species within the same sanctuary? So to introduce my study site a little bit to you, over here you can see a map. This is the Kayankarmi Marine Sanctuary. It is in the eastern coast of Sri Lanka. For those of you who don't know what Sri Lanka is, it's a small island in the Indian Ocean right under the Indian subcontinent. This is the Kayankarmi Marine Sanctuary. It encompasses 953 hectares. Something interesting that I want you all to notice over here is that this is the park boundary, and as you can see, it actually cuts right through one of the reef patches we see over here. So it was declared a marine sanctuary in 2019, so quite relatively recent. And the study site that we looked at looks at the sanctuary and also its vicinity, and encompasses a few fringing coral reef sites, which you can see over here in purple. Which you can see over here in purple and red. We also have patchy offshore sandstone reefs, which you can see over here in green. And I'm so sorry, I do not know why this keeps happening. Yeah. And over here we see the patchy offshore sand reefs, and in between we obviously have some sandy soft bottom habitats. So to delve a little bit into the tool that I'm using for my study, I'm using BRAVs, which is a relatively new tool that is gaining popularity because it's non-invasive, it's non-extractive, it's also relatively cheap, and it's very useful in cases when we want to observe the species without the need to capture them, for such as abundance studies which we are doing in this research. It's also very useful because it can generate quantitative data as well as behavioral information. And due to the presence of a bait, it increases the chance of fish entering the camera's field of view, which makes it easier for us to study. It's also useful because it overcomes limitations of different diver-based methods. So this is just like a picture of the Browse tool that we used in our study. So just to, this is just a schematic drawing of like the Browse, so you can see it a little bit better. And it usually consists of a galvanized steel frame, a camera over here, in our case, which was the GoPro Hero 7 that was set to these settings. And the camera is directly facing a bait arm, which contains a bait, which in our case was 1 kilogram of minced sardines. And there's a whole system of ballast weights, ropes, and buoys that are attached to make sure that it's on the sea floor, but also securely attached to the boat. So we did 52 deployments. Out of that, only 45 were successful. 7 of them failed due to either poor visibility, the camera not working properly, or really strong currents which toppled the Browse. So in total, we have 16 deployments in Kayankani Reef, 15 deployments in Elephant Point, and 14 deployments in Anemone City. There's a standard protocol, so each of the Browse were at the bottom for 65 minutes. And during simultaneous deployments, we ensured a separation distance greater than 500 meters to prevent any interference of the bait plume. So the next step of this was analyzing the video. For this, we used a specialized software called CGIS Event Measure. The species were identified to the lowest possible taxonomic level, and also during this, we noted down the substrate, which is either reef, sand, rubble, or mixed. We also noted down some additional characteristics such as the field of view, the bias, and the visibility when recording the videos. After the analysis was completed, we also carried out an agreement analysis to check for any observer bias. So these are just some reference images to see what the different substrates look like. So you have rubble, reef, sand, and mixed. So moving on to the data analysis, we carried out this at 2 different levels. The first one was family level, which looked at minimum count. This is not like a defined parameter that exists in literature, but we used it, which includes, so it is the addition of all maxen data of a species within a particular family group, so either groupers or snappers, within a single recording. And we carried out some exploratory analysis, Kruskal-Wallis and pairwise testing, PCA, and multidimensional scaling. And then we did species level analysis, which included maxen data. So maxen data, for those of you who don't know, is the maximum number of individuals of each species that is observed in a single point in a single recording. So for this, we carried out some exploratory analysis, principal component analysis, and then Permanova. So to go into our results a little bit, we saw 10 different species of snappers and 8 different species of groupers. And over here, you can just see what our data looked like for the different sites. And over here, you can see what the data looked like across different substrates. And I just want everyone to highlight this outlier that has really high abundance, which I will discuss in detail a little bit later on in my presentation. So we conducted Kruskal-Wallis testing. This is because we did pass the assumptions of ANOVA, and over here we see that there is a strong effect on substrate, greater than in site, and it was only the grouper distribution that was not significantly affected by the site. So then we went and did some pairwise testing to understand where the variation was coming from, and what we see is that Anemone City is significantly different than Kayankani and Elephant Point, whereas Kayankani and Elephant Point have similar abundance and distribution. And also what we notice is that the abundance in reef is higher than the abundance in sand across the species. So looking at the principal component analysis, we actually have like a really, I'm going to stand here, we have a really clear result. As you can see, that we have the clustering mainly based on the substrate. Over here we have the reef habitats, over here we have the sand habitats, and over here we have the remaining 2 habitats. Again, just want to point out that we have a really interesting point over here. And also within the habitats, we see that we have separation to a lesser extent by the site. So over here we have all of the elephant points within the reef, and then over here we have all of the Kayankani within the reef, and obviously Anemone City over there. And similarly across the different sand habitats we have. We also did multidimensional scaling. What we see is that the clustering is mainly along the horizontal axis, but we also see that there is greater heterogeneity among the different reef sites than we have amongst the other substrates we have. And again, just notice that point for data. Then we might go on to like our species level exploratory plots. And what we can immediately see over here is that there is similar richness of species across the different sites, but there's also greater similarity with Kayankani and Elephant Point compared to Anemone City, which is concurrent with our previous family level analysis. So this is a PCA wide plot over here, and over here you see this is the direction of increase in max N, and what you see is that substrate explains so substrate explains most of the variation across the first axis, and site explains a certain amount of variation along the second axis. And interestingly, species explains a very little bit of the variation, which then means to us that substrate and site actually have a greater effect on deciding the max N value compared to the species of interest. So this is our Firmanova. We carried out Firmanova for each of the individual species. I want to point out that we only carried out the Firmanova for species that had an addition of maximum that was greater than 10 to have a result that actually had some power and could be interpretable. And what we see is that for most species, that substrate and site do have a significant effect on the abundance. And over here, the sort of distinction between substrate and site is not that noticeable. So we don't see substrate having a much higher effect than site as we did previously. Also interestingly, Athalopoca roga was the only one to show site-substrate, significant site and substrate interaction. So moving a little bit on to our discussion, we see that in the family level that abundance in reef was higher than abundance in sand. This is pretty much as we expected and understandable because structural complexity provides shelter and niches and reduces predation, pressure and competition. So therefore it encourages more fish to gather in that area, which then encourages more groupers and snappers to go to that area. So what we know from literature is that increase in structural complexity increases fish abundance and richness. So we also see over here that there is higher variation among reefs, which again is understandable, and we know from literature that most studies apply a landscape scale perspective, but habitat can actually be viewed on multiple different scales. So when you look at the different reef habitats we have, they tend to be rather different from each other, with different coral compositions, with different niches, and different microhabitat structures, which then lead to variation in functional aspects such as prey availability and shelter. So again, once again, it's understandable that we have this level of heterogeneity amongst our reef sites. So interestingly, that one point that I showed you that had really high abundance, that corresponded to a sandstone reef. Unfortunately, it was the only sandstone reef that our brow landed on, so we don't have anything else to compare to. But we consistently in that video saw really high levels of fish, which is in line with literature because we know that sandstone reefs have really high biodiversity levels and they tend to be areas with higher numbers of larger fish, such as our groupers and snappers. So what this really tells us is that small discontinuous patchy habitats like Anemone City, which we sampled, can actually be really important for species survival and biodiversity. But what we see in Sri Lanka is that there's usually preferential bias towards coralligenous habitats, so they tend to be prioritized in terms of protection and MPA strategies. Oh my god, I'm so sorry. Something else that I really want to point out is that in Sri Lanka, the declaration and management of MPAs is really lacking in consideration of the unique ecology and the different species that exist, which is a really big shortfall that we have. And you can again see this when you see that the MPA boundary really runs through one of our reef habitats. So the recommendation based on this study is that we need to reevaluate the park boundaries of the Kayankani Marine Sanctuary and also preferentially include this Anemone City habitat, which has the patchy sandstone reefs that have really high abundance of groupers and snappers. So moving to my species level discussion, immediately as I mentioned, you can see slight habitat partitioning, but it's really difficult to analyze the differences at this stage because fish abundance and habitat is so species dependent. So because they tend to have different ecological preferences, we need to do a little bit more deeper. I need to go in and do a little bit more deeper analysis to understand where these differences are coming from. But what we do know is that Kayankani and Elephant Point are more similar in their species compositions than Anemone City, and this once again is understandable because they are much closer together They also tend to be more coregenous habitats with similar kind of structures as opposed to animalicity, and they're both fringing reefs as opposed to the offshore patchy reef that is found at animalicity. So like I was saying, this is also like an interesting plot because very easily you can see that the reef has a higher species richness, and in terms of groupers, we don't actually see any of them present in sand. And that is because groupers allow structurally complex reef habitats because it has an impact on their predictive effectiveness. But I need to go in and do a little bit more of a deeper analysis to understand the drivers between each of the species-specific preferences. One example I can talk about is the Cephalopolis algus is known to have strong site attachment behavior and usually prefers to exist in small core areas with high rugosity and reef complexity. But in our study, we actually found out that it is present across all the 3 different sites that we have. So I'm hoping to better analyze this in the next few weeks. But to conclude, I want to once again just highlight that these different patchy reefs, even though they're small and discontinuous, can be really important for species level analysis. And grubs is like a really important tool for us to understand the different spatial ecologies of reef fish, and hopefully we will continue this study for more long-term monitoring. Thank you. And
thank you very much for the presentation. Just in time, 15 minutes sharp. So now it's time for questions. First, the members of the jury are going to ask these questions. I don't know who wants to start.
Yes, sure.
Who wants to start?
I can start. Okay. Well, thank you. Very interesting talk. I have some questions about the first of all the sampling design. Did you stratify? How did you locate your groups, or just randomly? Or
how I did the drops?
no, I mean, how did you choose the locations? Because do you have the same number of drops on each type of habitat, or?
So yes, so what we did the drops per site, and the drops were sort of semi-randomized. We did have, because my supervisor was somewhat familiar with the area, he sort of knew some of the locations we should do drops in, but we didn't look at the substrate below. We didn't have an understanding of where the drugs were dropping because of certain limitations we faced. So in this case, our board, which had the echo sounder, which allows us to see the substrate, was not working. So we were kind of blindly dropping, and that's one of the limitations of the study, because we have really different representations of the habitat. So if you notice, the rubble and mix only represented 3% of our drops. And again, sandstone reefs was only 1 drop, which is a limitation, but we had no other option because we didn't have the capacity to go in and dive and look at the different substrates and drop the drugs accordingly.
Okay. So you were looking mainly at groupers and snappers.
Yes.
Groupers are very territorial,
Yes.
Compared to Lugenidae, and therefore you would expect that they wouldn't attract from a big area, whereas with the snappers, I can see that they would be attracted by the bait bloom from a larger area. How does that affect your interpretation of the results? Because you have much fewer groupers observed than snappers.
Yeah, I think that's a really interesting point because, for example, when you look at our sand, sorry, I just do not know how to make this work. When you look at our sand habitats, you can see that sometimes we do find the snappers in the sand, but we never find the groupers in the sand. So that's likely because even though they can smell the bait, they are more hesitant to leave their more territorial lands to venture further out within, whereas the snappers tend to be a little bit more inquisitive, so would leave. So that is definitely a probable cause and something to explore in the interpretation of the results.
And in the analysis, you use PCA. Have you finished? I mean, you didn't submit yet.
No, I haven't submitted that.
I would suggest, rather than PCA, to use redundancy analysis, which also allows you to do biplots and even triplots. And it's more appropriate for this type of data than PCA, which is more for mental parameters. So you still have time to do that.
Okay, yeah, I can look into that. PCA was kind of what they tended to do in the literature, which is somewhat the reason I did it. But thank you for
RDA or redundancy,
Yeah.
look it up. It's
Okay.
it's more appropriate for them. For
Okay,
this
thank
kind
you so much. I'll look into
data.
that.
Stop here.
Wants to go now?
So thank you for the nice presentation and congratulations for the work. So I have some questions about maybe to know your opinion about these techniques in comparison to others. So this week there was some other works related more with environmental DNA, for example. So in this case, what is the advantage of these techniques? I mean that each one is has different approaches and give probably different information, but at some point when you are just let's say presenting your data to managers or whatever is the responsible of managing these places, so how you can what is the advantage or disadvantage of the others against your product?
I think the primary advantage is looking at the context of where the study was carried out. So the study was carried out in Sri Lanka, which is a developing country with very limited resources. So the main advantage would be the cost effectiveness of the browse system and the sort of resource use that is available. So for things like environmental DNA or even less complex things like acoustic telemetry or like finding a species just requires a lot more funding, a lot more technical capacity, a lot more sort of resource use softwares and tools, which is just at this point not available in Sri Lanka. Even the study that I did is sort of most of the funding we get is for livelihood development projects, for management projects. And the research that we kind of do is built into these kind of projects. So maybe in a few years, hopefully with some grants or like a lot of development and research in Sri Lanka, I think we can explore other methods, especially environmental DNA would I think be something really interesting to look at and will give us a better, more holistic view of the species that exists within. But right now, at this stage of where we are, it's just not an option.
I think that the things have changed a lot because now just sequencing, massive sequencing, it's getting cheaper. So you will be surprised for that in the case. And another thing about this kind of, because you focus, as I understood, from the present ourselves of species. Okay. So if you want to apply for densities, so how valid are these methods? I mean, if you need to count.
Yes, so this method actually tells us relative abundance. So the max n value we use is gives us an estimate of the density that is there. Obviously, there is some amount of bias because we use a bait, and the bait tends to attract fish from surrounding habitats as well. So, but if you consider, but browse have been used to understand species richness, that have been used to understand biomass. Because right now I also used a mono browse system which only had a single camera, but there's the option to use stereo browse, which allows you to take measurements of the fish, so you can take length and weight measurements, and those can be used to estimate the biomass and the density of fish within. So that is something that is also going to be useful to explore in future studies.
For how long did you record? Because at the end, for me, I'm not an expert on that, but say, how you can avoid that you don't measure the same fish more than one time?
Okay, so that's perfect because that's a really good question. So that's what MaxN does. So for those of you who didn't understand, MaxN looks at the highest number of individuals of a particular species at a given time point. So if you're watching the video and we see Cephalopolis algus in one frame, there are only 2, and then in the next frame if there are 4, we only consider the frame that has 4. So what you're doing is you're looking at the minimum number of individuals that you are sure exist. So that way you're making sure that you're not counting the same individual twice because they're all on the screen at a specific given time. I hope that made sense.
Thank you very much.
So congratulations for such a nice presentation. Well done, even though it's not finished yet, but soon it's going to be over. So well done, congratulations. I think your presentation was clear. So I have some questions. I'm not an expert on this, of course, but I have some questions about this. Which is the effect of behavior in your way of sampling? Because and have you measured repeatability with this method? Let's say the same point. Have you measured several times and you're getting somehow similar estimates?
So we didn't do same points because the rub was dropped off in a way that would ensure that it's a little bit further away from each other to not have any interference with the blade plume dispersal. But what we are hoping to do in the future is to do a second round and third round of deployments. Unfortunately, during the 6 months, we were supposed to do multiple rounds of deployments, but we faced a little bit of an issue because the monsoons were going on and there were very bad weather conditions, which restricted the amount of times that we could do the deployments. But we're hoping to do more seasonally, so that will allow us to better understand the fish behavior. So I actually have a slide on the sort of limitations of the past study. As you can see, it's a really long list. And one thing that is not accounted for in this study is different behavioral considerations. For example, whether the species are nocturnal or diurnal, because some of the groupers also tend to be nocturnal, which means they're more active at daytime, dusk time, and evening. But what's interesting is because of the bruv is sort of more unbiased, it doesn't affect the behavior of the fish that much because there's no diver there, you're not scaring off the fish. And actually, the bruv is used to understand the behavior of the fish as well. So when I was watching the videos, I can note down things like whether it was feeding, whether it was protecting the bait from other fish, whether it was just swimming in the area, like whether it was unbothered by the fish. So you can also use the analysis to note down these kind of specific behavioral characteristics, which could be useful in understanding fish behavior as well.
Okay, and what about for how long was when you dropped that device? For how long?
So it's in the bottom for 65 minutes. But we discard the first 5 minutes when we're doing the video analysis to remove any sort of issues that might come up with the interference of us doing the deployment.
Okay, and all of them more or less about the same time of the, day. And which was the problem of doing like the same point several times? Or
it's because
it's not clear to me. So because it's not, okay. You're not really altering that much the environment, or what's the problem?
So what we do is we do simultaneous drops. So we go into the site. We drop one, then we go 500 meters later, we drop two, then we go 500 meters apart, we drop three, and then we go 500 meters apart, and then we drop another one, and then we go and do a round of collecting, then we go and do another round of dropping, and then another round of collecting. So if we were to do it in the same place, we don't know if there would be any, like, you know, some of the bait left over from
No, but I mean, I was thinking more like something like, okay, I will do it one run today, and I will, at some point I will be coming back in a different day and see if more or less I'm recovering some, because you might expect a lot of variation or not, I don't know. It's somehow going towards the repeatability of the method. That I'm sure it's already demonstrated, so
that's actually, sorry, I didn't understand your question. That's a very valid point. It was something we were looking to do, but because we didn't have enough field days to go in and do the deployments because of the weather conditions, we wanted to get more spatial coverage than to increase the sort of deployments in a specific spot. And especially because of the bait plume, the exact spot that you are dropping in doesn't matter as much as like the sort of area of dropping, because it attracts fish from outside as well.
Yeah, okay. Thanks a lot, well done, congratulations.
Thank you.
So thank you very much, Gonzalo. Any questions from the audience? Someone wants to ask anything? No? I have a question myself. And I see that you put there now in your study limitations. This is very important to show the limitations and to admit that your study is not perfect is good. Because you put there use of non-parametric tests affecting robustness.
Yes.
So because you use Kruskal Wallis.
Yeah.
And why did you use Kruskal Wallis?
So we did the testing to see, we tested the assumptions of ANOVA, and none of our data sets passed either the Shapiro milk test or the Devon's test. So it just, the assumptions of ANOVA were not met. So we had to naturally move to Kruskal Wallis. And over here, the limitations I've put are just limitations in carrying out the sampling itself. There are lots of other limitations that we face, but hopefully.
But how many data you have?
So I did 54 drops.
54.
So the idea was to do 16 per site. Only 45 of those drops were valid. So it was 45 hours of video footage that were analyzed.
But what I mean is that not only you, many of you have jumped too quickly to the non-parametric. You can use other distributions rather than normal and keep going with a parametric analysis. You can keep an ANOVA if you have a large number of data. And it's more robust, it's more powerful, and it's as robust against heterogeneity of variances as Kruskal-Wallis. So, but it's not only you, it's lots of things. So just to take note.
We tried some transformations, but it was always failing the test, so it just seemed like a safer option to do Kruskal-Wallis. But since the study is not done, I can look into it for the next.
Then you can change to another distribution like gamma or something like that instead of normal, and keep going.
I'll look into it.
Okay, so I think we can move on. Thank you very much for your presentation. So Carla is going to give the next presentation.
Can I just pass?
Yeah.
Okay. I'm gonna try first. Okay. So, hello everyone. I'm Carla, and I'm gonna be talking about the effects of Greenland's melting glaciers on macroplankton and trophic structures in 2 Arctic fjords. So first of all, it's really important to know that fjords are important ecosystems both globally because they act as carbon sinks and locally because they support productive fisheries. And as that, they sequester approximately 11% of the global marine carbon, and they support diverse population of fish, seabirds, and marine mammals. But because of global warming, it's causing an increased melting and an increased runoff. So this results in an increased freshwater input and stratification, which has implications for the marine productivity. And moreover, the marine productivity in the fjords depends on the type of glaciers that are modulating them. So for instance, we have marine terminating glaciers, which have subglacial runoff and also subglacial discharge and surface runoff. So the less dense water column goes up and brings nutrients from the bottom, which provides higher primary productivity and sustains abundant zooplankton populations. And then we have land terminating glaciers, which have rivers that supply turbid meltwater, and this prevents the mixing and upwelling of the nutrients, and then we have low light and stratified environments, so the primary productivity here is lower. But because we have an input of the open water masses in here, so at the mouth or outer part of the fjord, we have a higher productivity or availability of nutrients rather than at the inner part or head of the fjord because it's more affected by the presence of ice. So we would expect that with the climate change and the melting of ice, the marine terminating glaciers would transform and shift into land terminating glaciers. And because the phytoplankton are at the base of the food web, we would also expect to see these changes farther down the food web. So for that, we can use stable isotope analysis of nitrogen because it tells us something about the trophic level diversity, and of carbon because it tells us about the food source diversity. which can be represented nicely by plots. And it gives us the visualization of the isotopic niche, which tells us something about the resource and habitat use of the animals in the food web. And moreover, the isotopic niche is also a proxy of the trophic niche, which tells us something about the position of the animals in the food web. So it's represented by ellipses, and if the ellipses overlap, it means that the animals feed on similar resources, but if the ellipses don't overlap, it means that they feed on different resources. Moreover, the trophic niche also tells us something about ecological plasticity and trophic diversity. So this means that, for example, we can have an animal that feeds on a lot of different resources, so we call it a generalist, and if it only feeds on a little amount of resources or a really narrow range, we call it a specialist. So the aim of this study was to know if there were differences in trophic richness and density for macrobenthos and differences in trophic niche width for both macrobenthos and demersal fish assemblages between and within two contrasting fields. So for that, we wanted to solve both a land-terminating glacier and a marine-terminating glacier field, but because during the expedition we had an increase and unexpected melting of the ice, we ended up sampling Igaliku, representing the land-terminating glacier, but Tsunuye Creek had both types of glaciers, so it had a mixed effect. And we wanted to test two hypotheses. So the first one, we expected differences between the two fjords. So we expected less taxon richness and density in the fjords with land-terminating glaciers because of the less primary productivity that I told you about. And this would be also reflected in trophic niches that would be narrower in also these land-terminating glaciers. And then within the fjords, we would also expect something like that, but only for the land-terminating glaciers. So we would expect less taxon richness and density in the head because of the presence of ice, and this would also be reflected in narrower trophic niches compared to the mouth. But in the mixed fjord, we wouldn't expect that for the continuous supply of nutrients that it has. So for that, we went to South Greenland the past July, and we sampled these two fjords at the head and at the mouth. And for macrofauna, we used the box cutter, we received the samples, we sorted them on board, and then when we were back in the lab, we identified them individually, we ground and encapsulated them, and when necessary, we used acidification to remove the carbonates. And for the demersal fish, we caught them by land fishing, we identified them and measured them and weighted them. And then we extracted some a piece of muscle for the stable isotopes. And then at the home lab, we also ground and encapsulated them for the stable isotope analysis. So for the data analysis, we wanted to test differences in richness and density, in community structure, and which were the species or taxa mainly driving those differences. So in this case, I repeat, we didn't use a fish because simply the line fishing doesn't provide the whole or accurate approximation of the equifaunal community, but they were used in the trophic diversity, and for that we used trophic niches, as I explained before, and we used two different approaches, a classical one and an innovative one, which is based on the clustering of the isotopic signatures. And lastly, we also conducted taxon-specific analysis of the taxa that were driving the differences in community composition. And we also included these three taxa because of their relevance in the ecosystem. So Neptidae and Garrus macrocephalus are both predators of macrobenthos and demersal fish assemblages respectively. And this starfish is a mud-dwelling organism, so it affects the whole macrobenthic community. So for that, we use community-wide metrics, which is based on the calculation of standard helix areas and the Lehman metric, which these metrics tells us something about the spike occupied of the animals in the biplot that I showed you before, and the traffic redundancy within the food web. So our results showed us no differences in richness between the two fjords, so it refuses our first hypothesis. But then within fjords, we found a higher richness at the mouth, which confirms our second hypothesis. However, there were no differences in density between or within the fjords, which refuses parts of both our hypotheses. So how can we explain similar densities in both fjords? There are several reasons. The first one could be that the two fjord samples are not contrasting enough. Remember that one has the two type of glaciers, so maybe it is causing not as different results as we would expect. Then we only used box score samples, and during the campaign we used other devices, so maybe incorporating those other replicates would provide a better representation of the ecosystem. Then the communities might be just adapted to different conditions, and that's completely possible. And then it's also important to know that these analyses are based on the community level, and maybe the differences between two fjords are represented in taxon. So within fjords, yes, we found a higher richness at the mouth, but the density was similar, and it could just be because, yeah, we have more different species or taxa, but they have different contributions, and the overall density is the same. For community composition, we found significant differences between and within fjords, which was mainly driven by polychaetes, and so Oenidae was more abundant in the mixed fjord, and Sciocatepterus typicus was only found in that mixed fjord as well. And then Maldanidae and Spionidae were more abundant at the head of the fjords. However, these taxa are not the only ones that were driving differences in the community composition. So in the mixed fjord, as I told you, we had Oenidae that was more abundant there, but we also had this gastropod, and both of them thrive in productive variants. In the land terminating glacier fjord, we found this bivalve that thrives in low oxygen environments, and we had some results from the campaign that showed high sulfur reduction rates in the land terminating glacier fjord, which points at more oxygen depleted sediments there. So this would have like a trend of a higher productivity in the mixed fjord. So within the fjords, we found, differences in community composition, but we couldn't really see any clear trends. So incorporating environmental data from the campaign, like organic matter and pigments concentration, might help into creating this relationship with our results. And then for the traffic diversity, in terms of community, we can see also any differences between fjords. We can see like a rate overlapping the traffic niches, and this actually means that the communities in both fjords feed on similar food sources. But we also found high traffic diversity, which means that the communities there are specialized in different types of feeding. But overall, we can't accept our first hypothesis with this. However, within the fjords, we also found wider traffic niches at the mouth of the fjords, but in the mixed fjord, you can see that it's not that different. And the smallest standard ellipse area was found at the head of the land terminating glacier fjord. And all of this confirms our second hypothesis. However, this can be due to the fact that there might be indeed less food sources at the head of the land terminating glacier fjord, or that the community there just exploits a limited amount of resources. So this will be further addressed with mixing models. And for taxon-specific analysis, we saw that between fjords, most taxa had plasticity, so they fed on different resources. We don't see any overlap. But these three taxa were specialists, so they fed on similar food sources between the two fjords. They overlap a lot. Then, within the fjords, most taxa were plastic as well. And then we found higher isotopic signatures at the head of the mouth, which would point at niche partitioning or an efficient utilization of organic matter. But this will also have to be further addressed with the environmental data from the campaign. So overall, we can't accept our first hypothesis that between fjords there's less accent richness intensity and narrower trophic niches in the long-terminating glacier fjords. But our taxa supported that there are differences within the long-terminating glacier fjords and no differences in the mixed fjords. So in conclusion, I'm very happy because this is the first indication that even in small differences in glacier types in Arctic fjords, in the southwest part at least, there are differences in the food web structure. So just a reminder, between fjords we found differences in community composition that were not translated in trophic diversity. We found both specialization and plasticity in the taxa, and within fjords we found a higher richness at the mouth of the long-terminating glaciers, which was also represented in the trophic diversity. And most taxa was plastic. So just take-home message is that it's really important to understand how these glaciers and the presence of one or the other affect the fjord ecosystems in a context of global warming and glacial retreat. So that's all. Thank you for your
Yeah, perfect timing. 14 minutes sharp. That's okay. Perfect. So, who wants to start with the questions? You want to start?
Okay, so congratulations for the nice book. Okay, now just it's time for the discussion. So relax, and because you did a very nice and good work, plenty of information, data, treatment. So well done. So one curiosity in this aspect. Okay, so with this kind of composition, it looks like, or the impression at some point is that even within the fire, so you found significant differences into. At some point, it gives the impression that more than when you compare from one fire to the other.
Yes.
Okay, and so what's your impression of that?
Yes. So the thing is that we have a lot of data from the campaign, and everything points at different things. So it's not really conclusive. And the thing is that this is the first study that is carried in such depth. So it's really complicated to really make any strong conclusions about it. So I do think that because I only did part of the macrobenthos, which is like a very specific community in the fjord, and yeah, at the same time it's like a really general approach. That's maybe why I don't see any differences, because as I showed you, like the richness was a bit different, or not even different between the two fjords, but there were differences in community composition. So I do think that that's the most important key point, because the communities differ, even if the richness doesn't. So it's just that they contribute differently, or they, like in one fjord, there might be more species of one that might be more, I don't know, adapted to that fjord or those conditions. And yeah, that's all I think. Like I can't really make strong conclusions about it.
You say it's a preliminary study.
Yeah, it like
you're
this,
breaking the ice.
exactly, exactly. Like, so we went on the campaign last year, and these are the first results of everything. So it's really like.
What is the distance between one and another? you show them up at.
I don't know the exact
It's not much.
distance. It's really not much, no. Maybe, yeah. So, yeah, we wanted to sample this one.
The scale is at the
That's-
top.
yeah.
I know.
It's, yeah, so I don't know, 10, 15 kilometers.
And for example, this small compartmentalization that you follow in different niches according to that, so it was something that you were expecting from the beginning when you designed the sampling, let's say?
The thing is that when we designed the sampling, I mean, that was not one of the main things that were in the proposal of the project, let's say. But when I was looking into my data, I was just like, I think it might be interesting to see how specific taxa behave, because it's also important to know about the resilience of the community. So if taxa are plastic in that sense, it means that they can adapt to different conditions because they change their feeding behavior depending on the environment they are. So that's important in terms of climate change, let's say, because if in one period, now I don't have this data right now, but if in one period the temperature is slightly higher or lower, that would mean that they can adapt and thrive in different conditions. So that's really interesting, and that's why I wanted to look more closely into them, but it was not part of my first
And do you know if there are future plans, I don't know, just to do samplings or just repeat in different seasons or different?
Yeah, that would be really nice. I don't know. The thing is that the project is right in the middle, so they are gonna ask for also more time to process all the data and have like a more integrative approach of everything, because we had a lot of different things. And yeah, I mean, I do think that it would be really important to do another one, because this is really just basic study. So if we really want to compare, like for example, I was saying, yeah, this is the head of the fjord and this is the mouth, but really it's the head would be this and the mouth would be here, you know, but because of the sampling constraints, sometimes you're not sure, you can develop one device or another, so it would be really nice to be able to maybe go a little bit in and take really the head samples or out and really the mouth samples to really have these two contrasting points, and then just have like a better representation of our data. But it was really difficult because, yeah, I told you there was a lot of melting and our vessel wasn't made for breaking the ice, so at some point it was a bit scary.
Yes. Okay. Thank you very much.
Yeah, so thanks a lot for such a nice presentation. Well done. You're almost done. Almost done. So yeah, so I mean, of course, I'm not an expert on this, but do you think that has a huge impact related to what you said before? What's happening in the mouth? How that affects? So if you could do a different design, somehow you mentioned before, would you consider those? Because at the end you have two different kind of fjord, I understand. But then what's happened? Is that really affecting the productivity there? Or which is the overall, which is the importance, relevance overall? That's what I'm trying to ask. What's your opinion about it?
Of the mouth or of something?
Yeah, in general. So is that really changing? I mean, it's locally changing, definitely, but which is the impact that has in a broader sense? You know what I mean?
I'm not really, I think.
So
Just
you are seeing some local effects,
yeah.
and you are detecting some local effect, but the overall for the region, is that really changing significantly? Could you do some design in order to test how significant is that change? That's what I'm trying to ask.
Yeah. I mean, yeah, that's the whole point. So it was really complicated to know what my results were telling me because there are nearly no studies about this.
Right.
So the only studies that I found, that there are and that I based my my hypothesis.
It's not
is
critical, it's
different.
just I want
No,
you
no,
to
no,
know your
no,
opinion
no,
about
of,
it.
Of course, no, no.
What
and
would you do differently,
it's
or
completely fine, but yeah, like right now, there's only like the oceanographic and phytoplanktonic part. So indeed, like to know best if these changes are applied to the whole region, we should do more sampling in different fjords, in contrasting one or similar ones. Like that's the thing; it's so unexplored
okay.
that
I mean, you need to start from somewhere.
yes.
That's clear that you are settling somehow the basis for future work to compare with. That's clear and that's necessary. At some point,
Yes.
someone has to start. So in that sense, it's great. But what I was asking is more like a reflection from your part, as you were telling me, and you did. It's fine. It's fine. The other thing, correct me if I'm wrong, but. Yeah, when you were working with the stable isotope, yeah, the trophic web, so did you take different prey items and you validate that, which is the because sometimes happen like that, you know, that you might apparently think like, okay, they should have a distribution like that, they should have a distribution like that, but when you analyze the different prey items, they are given a different signal and they
Exactly.
magnify, so the effect could be magnified or not, and it's tricky.
And that's what I was trying to say here with the mixing model. So
Okay.
I didn't have this data yet, it wasn't processed, I couldn't really do it, and I didn't have time, honestly. But yeah, there's a PhD student, which is Hannah, and she's gonna do it, and
Okay,
I'm
okay.
sure she's gonna do a super nice work.
Okay, thanks a lot. Great job. Well done. Thank you.
Yeah, okay. Yes, can you tell me what time of the year was this done?
It was July,
July, so that would be like
in the summer.
the bloom, the time when there's a big phytoplankton bloom, and that's what's driving, I mean, it's the tritus or whatever falling down to the bottom that's driving the benthos.
Yeah.
But in that map of the sampling locations, are those numbers depths in meters? The sampling locations? Because it seems to be a big difference in depth between one fjord and the other one. This one's much shallower, that's
yes.
like 12, 13 meters, the other one is in the 50s.
Yeah.
Would that be
it's-
a factor in the community differences? Also, did they look at
Definitely, and even more like, I'm sorry, do you want to finish?
No, no.
Also, Igaliku had like a sill in the middle of the fjord, so that was also preventing the water masses from the outside to really go in, and that also made that some differences. So yeah, it definitely, like, the topography of the site also affects a lot, but I didn't find any differences in the macrobiotic community for that, or yeah. So
the sediment types were the same also on both?
sediment types, they varied mostly within the fjord. So it was almost muddy all along the fjords, but in the mouth it was a little bit more bouldery. Because yeah, like for example, in here there's like a little bit of a mound, or it's more shallow, and sometimes there are like deposition of rocks from years ago. But yeah, it was mostly at the outer part of the fjords where we found the boulders, and the rest was really, really muddy.
So, just to clarify, would you expect more of a terrestrial source of nutrients in the
From the land terminating glaciers, definitely. That's the whole point of comparing the two of them. Yeah, and that's why we say that there's less primary productivity because of this input of fresh water and the turbid meltwater, that it makes that there's no upwelling of the nutrients, then it affects the productivity in these fields.
I think what is missing is a sort of a multidisciplinary, taking into account everything, the currents, the oceanography, the primary production, all that kind of stuff to try to...
This is the project that I only did this. So, yeah, they are gonna do it, but I was the first one to present my results.
Okay, thank you. Okay, any questions for the public, please? No? Yeah, well, first of all, congratulations. I really enjoyed the presentation. I think in this plan, there should be somewhere an assessment of what's going on with the zooplankton, no? Because that's a very important link. Because some of the animals that you have there in the benthos, they do have planktonic larvae, no?
Yeah,
Some of
and
them. So,
we also have the zooplankton thing, yes. And they are working on that with a lot of super nice devices, like taking pictures and training algorithms and stuff, so it's really...
LOPC and these kind of things.
Yeah.
Laser optical plankton recorder.
Yeah.
Okay.
They are...
But what is your insight about them? Do you think that the planktonic, the zooplankton community is going to change a lot in between these two kinds of fjords? Any? Yes, just for you
It's
to...
less
to
abundant, that's the thing, that's the thing, it's really tricky because we had an internal meeting just before this, and it was really confusing because the zooplankton was less abundant in the mixed fjord compared to the land-terminating glacier fjord. So
Could be.
maybe it's grazed on more efficiently.
Or maybe it's because thermal stratification that sometimes makes zooplankton, facilitates zooplankton aggregation.
Yeah, that as well. They also found some differences in the type of zooplankton, so they have some like more active zooplankton, and I'm not an expert in zooplankton, but they were, they could see some differences in the mixed fjord. They were more stratified. Yeah.
And just, I wanted to say something that I think is very important. You are doing research in the most important place to do research now. And it seems that we are underestimating a lot the amount of ice that goes into the water.
Yeah.
When you compare climate models with reality, with satellite observations and measurements, it seems that we are underestimating that a lot.
It was so sad to see like how it was changing as we were approaching it. Like
we are in course of collapse with the Atlantic circulation, the North Atlantic current, and the AMOC, and that would be very. So what you are doing is very important. All we should be there doing research in Greenland at this point. So congratulations for that. So now is the turn of Leo. Whenever you are ready.
Yeah. Okay, so good morning everyone. Today I'm going to talk about my master thesis. So I work on the implication of omitting unclassified individuals from sex-specific growth models. I did this work with Professor Dr. Colin Minto in Galway, so at the Atlantic Technological University of Galway. So today we're going to talk about the growth of fish. The growth relates the age of the fish with the length. It's widely used in fisheries to calculate indicators or threshold for fisheries. Many different models exist to fit and study the growth, but the most used one in fisheries is the Van Bertalanffy growth model. It has three parameters: the asymptotic length, the growth rate K, and the theoretical age at which the length is zero, so the T0. The growth is different between fishes, and the parameters are also different because it's related to the life history strategy of the fishes. but it can also be different between the sexes in the same species. That's why it's important to study the sex-specific growth of fish. As you can see on the left with blue withing, the growth of male and females can be different. Please be careful because the females are in blue, so it can be confusing. So it is important to study this difference because for fisheries management, if the females are bigger, they can be more impacted by fisheries. For example, if the fisheries targets bigger fish, the females will be more impacted by size-selective gears, or maybe if some quotas are settled in some areas. And that's why it's really important to study this difference in growth. To study this difference, we need to know the sex of the fishes, and to do that, we need to open the fish and to visually identify the sex organs. So that's what is commonly done in fisheries. So you can see on the right here the sex organ of the females and of the males, but sometimes for really young and immature fishes, the sex of the young fishes can't be visually identified. And that's an issue because we are omitting a part of the data set and the sample. As you can see on the right, that's are the species I studied, and that's the sex distribution at age 0, so for really young fishes. And you can see that the proportion of unclassified individuals is important, especially for herrings. Be careful because these values of male and females, the distribution of male and females are stacked, and yet the distribution is along the length. So unclassified individuals are typically smaller than the classified individuals. So my research question is, what are the implications of omitting this part of the data set, the unclassified individuals, in the sex-specific growth modeling? To study that, I use the methods to include them in the sex-specific growth modeling, to guess their sex. So I used this paper from Colin Mito, my supervisor, that developed a method, statistic methods, to guess and to predict if the unclassified individuals are more probably male or females. So I tested this method for the first time. It's never been tested on real data sets. So I tested this method on 4 different species. I chose species with sex dimorphism, so with difference in the growth of male and females, but also with unclassified individuals in the data sets. So this method uses the EM algorithm. So the EM algorithm is the expectation maximization algorithm. It's a mathematical tool that can predict subdivision and cluster inside the data sets. So as you can see on the right, I defined 2 groups, males and females. These are simulated data, so yeah, just so you can understand. And as you can see on the right, the EM algorithm can predict and cluster these 2 groups. In my data sets, it's a little bit different. As you can see, it's difficult to see the 2 groups in the unclassified distribution. The unclassified distribution is normal. So we have to force the organism to take and to consider the sex ratio. So we use the mixing proportion, which is a tool that something that we can calculate before to say more or less what we expect for the proportion, and the algorithm going to use it to determine and to predict the sex of the fishes. So here, these are the predictions. So for herrings, I choose the example of herrings. So for herrings, the growth of females, the females are bigger in general, typically for the growth model. So the EM algorithm going to take that into account, going to take the classified individuals with known sex into account, and going to predict for the unclassified individuals the biggest individuals to be females, and the contrary for males. As you can see with the line here, that are the probability of with the length of the unclassified individuals to be males or females. So now we're going to focus on the mixed proportion. I already told you about, so it's the expected proportion of females among unclassified and classified observations, and there are several ways to calculate it. The first way is just to give all the freedom to the algorithm and to say, but the algorithm going to just this formula and this method is going to lead to extreme results because males are typically smaller and unclassified individuals are typically smaller than the females. So the EM algorithm is just going to say, well, they are all males. And that's not what we want. So we don't think that's reliable and realistic. So we choose to use the sex ratio of the classified individuals, and that leads to more reliable results, we think, closer to 50% with the probability of being females. So that is completely reliable, but we chose to use the last one and to give some freedom to the EM algorithm by also including the predictions. I can come back to it if it's not clear. So what are our results? Here you can see the growth model and the fit of the growth. Here you have the age of each species and the length. So it is not clear to see the difference. The unclassified individuals represent a really small part of the data set if we compare to the whole age. But if we zoom in here, you can see that the growth curves are pulled down by the EM algorithm. And there is actually a difference. Yeah, you can see the EM algorithm, in dashed line. And the FMU method, so the FMU is the female, male unclassified, so it means that we omit these unclassified individuals to calculate and to fit the growth model. So we also studied the impact on the growth parameter. So here you can find the growth parameter. So the growth rate K, the asymptotic length L infinity, the theoretical age at which the length is 0, T0, but also the standard deviation to study what's the deviation for the dispersion of the data around the growth model. So what we can say about it is that the EM algorithm mainly impacts two parameters, the growth parameter and the intercept T0. You can see in green, I created a statistical test. I need to work on it again if I want to publish it, but I created this test to see if the difference can be considered as significant. So for the intercept T0, all the differences are significant. For the growth rate K, it's not the case, but you can see that we can have, there is the same trend and the growth always increase, the growth rates. So yeah, the increase in the growth rate K makes the slope steeper, the slope of the growth curve steeper, and the intercept T0, the increase of the intercept T0 going to pull down the curve just a little bit. So yeah, that's our impact on the visual impact on the growth curve. So, what do we have to think about these results? I think that I demonstrate that if we omit unclassified individuals, we are making errors. We are omitting a part of the data set, and we need to include them to avoid bias. Fisheries management and the fisheries field in general is very important because it impacts a lot the ecosystems and the stocks. So we need to be precise because all the fishing pressure, all the threshold value, the quotas are calculated using indicators from biological indicators such as the growth, but also the spawning and other values like this. So we need to be precise. And this algorithm allowed us to be more precise on these important values. But I also discovered huge potential for these methods. I think we could use this method for fully latent variables, variables where we can't, where we don't have the sex observation, where we don't know if the fishes are male or females. We need to do more work on it, but I think that's very likely to happen, and I think we have the knowledge to do it. If we can do it, it means that we don't need to open the fish and to visually see if they are male or females to classify them. And that can be a huge step forward for a non-invasive survey where we don't need to kill the fishes. And that will be combined with other technologies such as AI, such as intelligent trawls. And I think for this type of survey right now for fisheries management are really expensive, and that can help a lot to get more data for fisheries, because fisheries management and biology in general can be difficult to find data, and it's a data limited area in general. So that can be really helpful for science. I want to also quote this. So mathematics is biology's next microscope, only better. With microscope, we can see the really tiny cells and tiny things, but with mathematics, we can see the unobserved. So I think we should look into this type of algorithm and this type of science, combine sciences for science. So I would love to finish with this sentence, but there are some limits for this model. So first of all, we are clustering and subdividing a data set. And if we want, for example, to divide a fully latent data set with no sex observation, we need to be sure that we are subdividing the sexes. Because, for example, the cohort could have different growths, and also the gear used for the sampling could lead to different results in the growth. So we need to be absolutely sure that we are subdividing the growths. We also need to do more work on the mixing proportion, for example. These results, I think, these methods, the classifying global methods, I think are reliable, but we could maybe look into the spawning sex ratio, or maybe the sex ratio at young age, and not take into account the full of the ages. And yeah, we could do more work on that. And also maybe on the statistical tests, because of course we are predicting the sexes, so that leads to answers some errors, and that needs to that's need for the works. And I think that's it for me.
It's time for questions now. Who wants to start?
Okay, so congrats for the work. I think you explained really well, so and I enjoyed that. I think that it's a necessary research. Because, I mean, I'm not an expert on that, but I think it's quite important to see how you can improve and increase the knowledge for management of fisheries. So I think your whole research is towards that direction. So I congratulate you for doing that, and I think that it's not an easy task, and you explained it really well. So well done. So just correct me if I'm wrong, maybe I understood wrongly. But this FMU versus EM model, yeah? But in any case, what is not clear to me, I understand that what you did, imagine, I know for certain size classes, yeah, I know the sex, so I can even use that proportion to help to algorithm to split them, no?
Yeah. So that's a really good question. Maybe I was not clear enough on that. So
my question is more really, maybe I lost myself in that. So which is the improvement between that and the other assumption that we are doing? You know, in the global, when you are talking about the global, can you go to the next, I think, towards the last slides.
Um,
Would you have, yeah, that one, yeah? Would it, so which is,
yeah.
when you show us the improvement, that is including which, doing with global or with classified? That's my question.
Okay. So I'm using the global equation.
Okay,
I could use the classified equation. As you can see, the results here are like the mixing proportion is not very different. So it will lead to very not a lot of change in the results. So it will be fine to use both. We choose to use the global equation because we saw that maybe males are more likely to be classified, because we think that the fact that we can't classify individuals is related with the length, and because males are mostly smaller, maybe they are more likely to be unclassified. That's our guess, but we need to do further.
so it was not clear to me, how do you decide, which metric you use to decide if it's classified or global, which is better? That was not clear to me.
Yeah. So
How you test that, you say, no, you know, the proportion, which metric you use to decide between those two?
So it was only visual. I actually can't say that this method is better scientifically. I need to maybe do some genetic research to see if the unclassified individuals are male or females. But for now, we just chose that a result close to 0.5 is better, seems more reliable. But that's why I'm talking about it in the limits. We need to do further work on it.
Okay, and in terms of, if you have to add new species or other species, in which species you will focus on?
I would focus on.
And what you are gaining for that? That's it.
That's a good question. I didn't talk about it actually, but I think I would focus on species maybe in areas where with limited budgets, so we can, or with limited knowledge, so we can have more data easily and maybe help these areas. So, and where also the fisheries management needs more information, more
Like
data.
data fisheries.
Yeah. I think it's not really, the importance of this study is not related to different species, but more with the context of fisheries and this type of studies.
Thanks a lot for that. Great job.
Yeah, that's very interesting work, very original. So this is based on the assumption that this mixing proportion you get from the ones where you can determine by opening them, you look at them whether they're male or female, and that's your mixing.
Yeah, that's right.
And you're assuming that in age 0, that is the same mixing proportion, right? It remains constant.
Yeah.
Okay.
For the classified individuals, not for the
Okay.
because there is a classified individuals at age 0.
Okay. But okay, so if males in general are growing slower than females, and we know that there is size-selective predation, for example, size-selective mortality, then I would expect during age 0, the proportion would probably change. It would not be constant for the whole age 0. So, but you are considering a constant proportion during age 0, right? So maybe that needs to be to be considered. Okay. So, in fact, most when you see look at most age and growth studies where they fit a vertically model, they don't even have age 0 fish because it's always the problem of sampling.
Yeah,
so
actually I don't really know what does mean age 0. I had data with age 0, but I was surprised how, because for me age 0 it means that they are born this year. So yeah, I don't know.
that's because of selectivity. Usually we don't have the data for age class 0, and that's why the growth curve, you have T0, the absolute value of T0 is quite big because the curve, instead of going towards 0, it goes further up. So if you can do this, this would be a big improvement to the fitting of the model. Also, did you in the literature come across stochastic growth models where they assume that the growth parameters have a distribution?
Yeah, I looked into a lot of different studies about growth. To be honest, most of the time it's difficult to see because it's a mathematical language and I'm not used to read this type of article. So I tried my best. I did not understand everything, and I also tried to do the statistical test, but it's very difficult. It was very difficult for me to.
No, I'm just wondering because there is a stochastic convertible anti-growth model where each parameter is assumed to have a distribution, gamma distribution or normal distribution, and that kind of maybe if you do it for males and females separately, may solve the problem.
Yeah. Do you know what's the name of the paper?
Yes. It's a Russian. The author is Troikinov and something. I can look it up.
Thank you, congratulations for the great work. And so, as my colleagues already say, so it looks promising as a model in order to. My question is more because when you talk about the application in fisheries, so I show a very useful application in the case of in order to understand the biology, the growth, and so on. Usually, when this kind of strategies. So they focus more on the reproductive ones, which are the ones that, for example, if you want to predict the future biomass for the next year, even in order for establish the quotas and so on. So what is in order to know, I don't know, how many female males you can have in very juvenile ages, so. And as Karin said, so then you have selection prediction in early ages, and so maybe according to the size one of the sexes it's more related than the other. So what is the advantage for a future, let's say, biomass estimation or so on, and reproduction in fisheries?
I think that it's almost impossible to have a perfect fit of the growth, first of all. So I think that's just a step forward toward more preciseness. Because we are, like right now, for some stocks, we are augmenting a part of the data set. So it's just a step forward. Then maybe we can use that on different models to compare the models. But your question was more on the maybe what would be the impact on fisheries management, and if we combine it with the sex spawning rate. Yeah. So we used, I think, for many stock we use the yield per recruit model. That is a model that use the growth, so the K, and the weight infinity, but it can also be the length infinity. And we also used the biomass per recruit. I think we also used the spawning per recruit. So I think for this precise example, it will only be beneficial for the biomass per recruit, yield per recruit parts, but yeah, I think I don't actually. So
There's no one else. No. I was thinking. I was thinking that probably there are two different estimations in this way. I mean that one thing is that what you expect according to the population composition, how many of them can reproduce, and so they use of whatever and recompose or the main composition of the population of the fish at the end. And then what I see very useful is in order to understand the biology and the behavior, in order to, because we face similar problems when trying to identify the sex in very early stages, and especially in some species where it's not so clear the sex, and so it could be a model that could be very helpful, especially with when the new, let's say, individuals arrive to a given growth area or whatever.
Absolutely.
And the estimation.
I was surprised that this model was not used yet so much in biology, in the literature. Maybe some other methods are similar to this one, but maybe with another name, and I couldn't find the papers. That's also possible. But I know that they are more or less the same methods, not the same, but the methods to guess the age for fisheries, the Batashivia methods. I'm not sure which one, but Yeah, I think this type of methods are used in fisheries because I feel like the connection between mathematics and fisheries are stronger than the connection with mathematics and biology in general. So that then could be applied to a lot of other fields in biology. Absolutely.
And you have studied different species.
Yeah.
Okay, and maybe you mentioned or I missed, so you found any kind of, let's say, differences or in complexity when applying the method depending the...
Yeah. So first, oh wait, I don't use this one, this slide. So the proportion of unclassified individuals is different between the species. Yes. So the impact is different. And if you want, like if we talk about the issues I have with this EM algorithm, for example, for haddock, I assume that the distribution was normal along the growth model, but actually it's not. It's log normal. So that's an example, and I should develop the model also to include log normal distribution along the length. So that's one of the issue, and also for the same species, the sex differences were not that clear. So that also can be applied.
Yeah, can I just?
No, thank you. No, it's okay.
No, because you brought up something. I just thought if you had a good sample of the size frequency distribution of H0 fish, and you were certain they were the same cohort, and there were differences in growth between females and males, then you could use, you could decompose the distribution like what you said, the Bhattacharya method, assuming, and then you would have this problem of worrying about the proportion because you could do that.
Yeah, absolutely. Yeah, I think we have a lot of work to do on this proportion in this parameter especially.
Okay, thank you very much. Any question from the public? Yeah, I have one myself. Well, first, congratulations for this work. I think you have explained everything pretty well. The complex stats you have taken your time to explain it properly. Very, very well done. And one question. How will your model cope with these trends that we are seeing now in some species of small pelagics? That it seems that they are reaching a smaller size, a smaller maximum size, maybe because fishing pressure, but also maybe because of the increasing sea surface temperature. Will the model account for that along a extensive time series?
I think right now no, but I think it would not be so difficult to take that into account if we want to develop the model. And to just, I don't know if it's completely answering your question, but I think we're going to have more and more unclassified individuals along the years because the young fish is going to be smaller and smaller. So, and I think the classification of the sex is related with this, with the size of the fish. So maybe it's going to be even more useful in the future to use this type of algorithm.
Okay, thank you very much. Okay, I think we are over here. Thank you very much for assisting this session. See you up here. See you around.
Alright, welcome back everyone. We are going to start the last session of presentation for this year IMDRC symposium. And so as usual, our students have 15 minutes to present their work, and then we'll have a discussion with the members of the jury. So first up is Elena, and Elena, whenever you are ready, you can take it up.
Okay, welcome everybody to this last session. I'm Elena Esteban, and I'm going to present you my thesis on the supervision of Professor Carl van Collen in the Marine Biology Lab at Ghent University. So first I'm going to introduce you the topic. So we all know that anthropogenic activities have big impacts on coastal habitats, such as beach dredging, sediment extraction, beach nourishment, and also organic loading, pollution, and this climate change effects like sea level rise or erosion. These environmental changes, of course, have effect on the macrobenthic fauna of coastal lagoons, and the coastal lagoons are particularly affected by the sediment deposition effect. So I'm going to present you the case study. I worked on the Zwin Tidal Lagoon. This lagoon is located between Belgium and the Netherlands. It is particularly important for wading birds such as avocet, storks, spoonbills, and it's a Natura 2000 area under the Bird and Habitat Directive, and it's included in the Ramsar list of international wetlands since 1986. Still, with this protection, the lagoon is gradually infilling with sediment deposit, and this has the effect of elevation of the land and reduced time of inundation that lets the breeding islands of the birds be accessible of terrestrial predators and reduce biomass of their prey organisms that are macrobenthic fauna. So, because of this, the lagoon has been under restoration efforts by the Flemish Regional Nature and Forest Agency. They have, in 2019, transformed this former agricultural land into tidal nature and expanded the protected area to today 333 hectares. They target actions to maintain desired sediment conditions for long-term conservation for birds and nekton and macrobenthic organisms. So why did I study macrobenthic communities? Because they're the most prey organisms of the birds. Macrobenthic species are the organisms that are above 1 millimeter size and that live in or on the substrate. They're really correlated to certain sediment characteristics in their environment, and it's important to study them to identify the optimal conditions so that we can guide the conservation and restoration efforts in the area. So those are mainly my thesis objectives. I want to study macrobenthic communities in the Zwin intertidal lagoon. I'm gonna analyze the spatial distribution of them and check the environmental drivers by doing multivariate analysis. And then I'm gonna model optimal sediment conditions of the important macrobenthic species with nonlinear pointal regressions. Onto the material and methods, the team sampled in autumn 2023 66 cores on the whole area to have a representative coverage of all the variable habitats. The samples were then sieved in 1 millimeter mesh size and preserved in 4% formaldehyde until the sorting. For the sorting, we stained the samples with rose bengal, and the macrofauna was manually sorted and identified on the stereoscopic microscope with identification keys and literature to the species level mostly. We took pictures to have a detailed data set with also ecological information that might be relevant for further analysis. For the environmental data, we had sediment cores that we took very near to the biological cores, and these sediments were analyzed with laser diffraction with the Malvern Masterizer 2000 and categorized with the Wentworth scale. our environmental drivers, our environmental variables are the median grain size, the sediment sorting factor, the mud content, the total organic matter, and the immersion time of each sampling point. For the statistical analysis now, first I started with some data cleaning, so removing the terrestrial species that we found in the samples or the hypervented species. We also calculated then the Simpson diversity, the abundance, and the species richness of each sample. Then for the descriptive multivariate analysis, we performed cluster analysis with break distance, and once we had those community groups identified, we then characterized them using Kruskal-Wallis comparison with, we characterized the environmental variables of each group. We then modeled the species abundances in function of environmental drivers that we identified in the literature, being the most contributing factor of their presence or absence. And we compared briefly the old and newly restored area in the Zwin Lagoon, and we finally mapped the dominant species in each sample group. Now the results. So first, some characteristic of the macrobenthos in the Zwin Lagoon. You can see here the most present species in the total of samples. Other species contributed to less than 25% of the presence. We found a total of 24 macrobenthic species and a mean richness per sample of 4.8. There were generally a very high variability in abundance and species richness between our samples. For the community structure, so these are the cluster analyses that we plotted on a non-metrical multidimensional scale plot, and we colored each sample group, each sample with their group. So what we have here is that the group 3 is characterized by high abundance of Aphelocheta marioni polychaete and a coarser and homogeneous sand sediment. Then we have the group 4 that's characterized by Edicea versicolor, also a polychaete, that is much more tolerant to variation in the sediment, and we can see that it's also correlated to very high sorting factor. Group 4 is here. We have the group 5 that has very low abundances and richness and no distinguished species, and it's very spread around our graph. The group 6, located here between the 4 and 7, has no common species, only the common species and no significant variable distinguishing it. And the group 7 here is characterized by very high abundances and richness, high median grain size, and higher immersion time than the group 4. This was the only significant difference for immersion time variable was between 4 and 7. For the models, so a quantile regression, nonlinear quantile regression model was applied to our two axes. So we checked the model fit with the residuals plots and the archaic score. And this is the Scorbicularia plana model against the mud content of the samples. And what we can see is that there is an exponential growth of the Scorbicularia plana abundance when we reach around 50–60% of mud content in the environment. For Edicea versicolor, the best fit model followed Michaelis-Menten function with the organic matter, and what we can see is that there is an exponential growth, and then it plateaus at around 2 or 3 organic matter, percent organic matter content in our sample. So briefly, to compare the old and new preserved area, we can see that there is very low overlapping between the two communities. This indicates that the two areas have different community groups. And when we ran a simper to see where this the similarity came from, we mainly saw that Ablatenuis at Bayval characterizes the new area, and Aphelocheta marioni, a polychaete, characterizes the old area. We found no significant differences in abundance, species richness, and Simpson's diversity between the old and new area. We map here the dominant species per sample in the Zwin Lagoon, and we have clearly some hotspot of preference habitat per dominant species. Note here that dominance was the number of individuals per sample. I know that biomass would have been a very good tool to have abundance value, but we didn't have time to weight all of our samples. So to discuss our results, we find that the findings are very consistent with other coastal lagoons of the area and with very diverse benthic communities in function of the habitats. We have dominance of Edisted versicolor and Epelomastis filiformis that aligns with their known preferred habitat that are muddy and sandy flats. We can see the presence of Scorbicularia plana, bivalve, and Aphelocheta marioni that require very specific habitat conditions, and that shows that the Zwin Lagoon has some habitats that are very well defined, as the map showed. For the group, we can discuss that the results by saying that the group 3 dominance by Aphelocheta marioni is mostly explained because of their need of sand for building the tubes that they live in. Edisted versicolor thrives in a very organic rich environment, as was shown in Davy and George's publication. And this species adapts to very diverse conditions. That's why we see it in all over our samples so frequently. The group 5, as we saw, was the one that was very spread, likely represents all our samples that didn't fit in any other of the groups. The group 6 suggests a transitional habitat between the 4 and 7, because it was really located here, and it has a mix of the two conditions of the 4 and 7. And for the group 7, what we see is that maybe the immersion time has a crucial role in the distribution of macrobenthic species, but here the data we had was sent by the laboratory of biology neighboring us, and this data lacked a lot of precision. It was more of a guessed immersion time than a true measure in field. The nonlinear regression models align and are coherent with our SimProf analysis results and with the literature that we found. When compared the old and new area, we found no difference indexes after 4 years of colonization, which means that the colonization process is really good process now. Although we have to nuance our things because we have very low sampling points and the new area compared to the old area, so 25 against 40 samples, 41. For the spatial distribution of the habitats, our map shows clearly very strong habitat types in the Zwin Lagoon, and that's more or less the discussion part. I'm going to conclude, but before I'm going to thank the Ghent University, the master, the Erasmus program, and my teachers, family, and friends for their support. This is the link to my reference list if you're interested. And to conclude, I'm going to talk about the restoration efforts. So my study concludes that we would recommend focusing on very high mud content and fine sediments in the Zwin Lagoon to maintain the prey organisms for the birds. And then for overall community diversity, the coarser sandy beds that are very sandy would support the species like Aphelocheta marioni. For future research, it would be very interesting to have additional environmental variables, not only sediment, but water characteristics, inundation time, that is way more precise at what we got. And long-term monitoring, I refer here to seasonality, because we only sampled in autumn, and maybe this has an influence on the distribution of our macrobenthic communities in the lagoon. In general conclusion, I think that this study helps to guide the effective conservation strategies that are ongoing in our Zwin Lagoon. I thank you for your attention.
Thank you, Elena, and as I admitted ago, so very good use of your time. So I'm going to ask the jury members to start the discussion. Who wants to go first?
Can I go? I'll go first. I have very simple questions.
Yes.
Well, I think you had a very nice presentation. It went fast sometimes. It was hard to follow.
I have one methodological question that has to do with your environmental variables. So you used the variable to characterize the sediment size, use mean size of the sediment.
A median.
Median size of the sediment.
Yes.
Do you think that given the importance that finally you acknowledge in the end of having very small sediments, something like the proportion of small sediments would be a better indicator than the median, because the median loses a lot of information on the structure of the sediment?
Yes, that's why median grain size was the most significant variable,
Yeah.
so that's why I presented this one. But of course, we took into account the sediment sorting factor
And the mud content.
and the mud content. And sediment sorting factor is a ratio between the 19th percentile and the 10th percentile.
Okay.
That gives the information of how homogeneous our sediment is. And of course, it's really important because some species tolerate very strict conditions. This sorting factor was not the most significant, as I think that mud content was so spread around our samples that it might influence way more than our sorting factor.
Okay. Now, the other thing is a detail. You presented the multidimensional scaling as the product of a cluster analysis. Am I wrong?
It's a representation of my cluster analysis, not the result.
How did you do that?
So I plotted just my macrobantic data on the multidimensional scaling, and then I said to the program to color my points with the groups that were assigned to them with the cluster analysis. Is that
okay?
Clear? Yeah.
I'm not very sure how you did it, but okay.
So it's a cluster analysis that each sample got 1, 2, 3, 4, 5, and then just a plot like how it's.
Okay.
And then it's
So you use the colors of the clusters on the graph,
Exactly.
but the graph is multidimensional scaling.
Yes.
Is okay. All right. The way you said it, I thought this was the result of
No,
the
no,
cluster
no.
analysis.
It's a
Okay, it's just the
representative.
coloring. Okay. Fine. I don't have any more questions. I think you did a very good job.
Thank you, Elena. I have a first question regarding your sampling design.
Yes.
Can you maybe go back to this map where you show it?
Yes.
How were the sampling stations decided? Was it based on special hypotheses? I know you didn't participate.
Yes, sadly I wanted to, but I couldn't skip the program to go sampling with them. What was explained to me, so this study has been taking place since 2010, and they sample every 3 years first and now every year, and they try to sample in the exact same coordinate position as the 2010 data to have some following up on the sampling points.
Okay, do you think there will be some modification to be applied to this sampling design in the future if it's a long-term survey?
So if it's a long-term survey, I would take into account that these inlet channels are very variables with the tidals.
Yeah.
And so sometimes instead of sampling exactly the same coordinate, I would sample where the inlet channel actually passes and not if the water differs from the position.
Okay. Don't you think it would be a good idea also to include more samples in your area?
Yeah, I also agree with that.
And then there's one sample that seems to be in the middle of the field here.
This one?
Yeah.
Yes. So I got the coordinates on a paper sheet that was stained with sand, mud, and water, and I had to implement them on my data, my Excel file, and I could not invent the actual true position of that sample. We imagined that it should be here. But
just nearby.
the coordinates are wrong, and I don't not gonna change a 5 for a 2 coordinate.
Okay, yeah. Well, it
Get
makes
right.
sense. It's 11. It has to be next to 12 or something.
Also, this one, for example, should be around here. So
Okay,
yeah.
interesting. Okay. I have a question about your quantile regression. I'm not very familiar with it.
With that?
The quantile regression
yes.
model. I'm not very familiar, but do you have like something that would like a correlation coefficient and some indicator of the percentage of variance that is explained, or do you have a p-value that was?
Yes, we have p-values on the model fit. So
Yeah.
if it truly shows trends of the lines. So what I can say is that for the blue line, the median line, the p-value was significant,
Okay.
but on the other lines, it was near, but not really. But anyway, the quantile regression is more of a visual to see how our abundance will change with a gradient of parameters.
Okay.
So the dotted black line is a basic linear regression that we all know to have a comparison. And what we can see is that, for example, here the linear regression would have overestimated the overestimated the abundance of helicity versicolor for 0% of organic matter and underestimated the abundance of helicity versicolor with a 6% or 8% of organic
But
matter.
is the variance explained lower for linear than for this one?
So when we check the model fit, the score, the
Was a better
IAC
Okay.
had a better one with the.
Okay. So you said it's exponential on the left and then Michaelis-Menten on the right.
Yes, more or less.
Is it just empirical or is it supported by some
So-
biological meaning related to this kind of model?
To build this model, I was alone to
Yeah.
do all my data analysis, and I tried to find references on which would be the easiest. I had some struggle to find the perfect function,
Yeah.
so I modeled various functions, and it was more of a try and error.
Well, you know why
method.
Michaelis-Menten kinetics was developed, right? It's basically to describe the
Epistemology.
enzyme
no,
kinetics.
I saw it in multiple papers for other species.
Yeah.
So I said, oh, why not try this one as the other ones
Okay.
didn't match.
My recommendation for regression would be, if you don't have a valid reason to believe that the relationship is exponential or like that, probably better just to use a linear one, which does not involve some assumption.
So it was one of
Even
my.
though I agree that the fit might be better, but you know the better fit is always with a high polynomial, and we don't use high polynomials because it doesn't mean anything. And that's why sometimes, even if it's not the best fit, the linear one is probably better.
It was one of my challenges and objectives to perform this nonlinear modulation because we're working on a paper with my supervisor and this paper contains nonlinear regression, so I wanted to challenge myself to try and build them also to gain knowledge with the thesis. Because I know how to do a linear regression.
Yeah, I'm sure you do, and you did one actually. And so you choose based on the literature to model the abundance of scorbicularia and helicity against organic matter or mud content. Don't you think this approach of a priori eliminating any other explanatory variables is not a risk of
forgetting
not just performing purely confirmative science?
Yes.
Because in the end you say, yeah, there's a relationship between mud content and scorbicularia. Yeah, you know that. You already know that. And the same for helicity.
Yes, I agree with this. To be honest, I run on air the models and I change the variables each time to see if I found something new, because if I found something new, I would have presented it. I didn't find anything.
Yeah, but you know, a variable can act individually or they can act by interacting with each other.
Yes.
Did you think of maybe running some model that would include several several variables?
I did think of that. I was very limited by time at this point of the data analysis, so I had to concentrate on one variable, one species.
Okay. Okay. So you end up in your conclusions talking about the habitat values for wading birds.
Yes.
How do you transfer your study into an information that is usable for conservation, and especially with the perspective of wading birds?
So the report will be sent to the agency that's regulating the area, and they have already put in place some path for the people that walk that will only be accessible to protect the islands, the mud sediment that from the crushing. So in a very concrete point of view, I think the job is now to identify where those high mud or very sandy are in the field, and then preserve this from.
Okay, but why do you think those habitats are better for wading birds?
Because the food, their food lives
What is it feeding on?
They're feeding on Scrobicularia plana, Eriseri versicolor, Heteromastus filiformis.
Does it feed on Heteromastus?
It's
It's a small species.
yeah, maybe not, but I think
So we're wading birds, I wouldn't bother targeting
so-
Heteromastus.
Scrobicularia plana for
Yeah,
sure,
definitely.
and Arenicola marina too.
Arenicola. Did you have Arenicola?
We found Arenicola, we counted them as individuals per meter square in the sampling point, but they were really minimal in our samples.
Okay. Yeah, I didn't see a habitat map. You showed a map, but it was just elevation.
The first one was.
So I guess
elevation.
it looks like you have salt marshes.
Mm-hmm. So there are salt marshes, there are just water canals. These are, we don't see clear, artificial breeding islands that were built for the birds that are covered in sand. And so you mean a habitat map with very defined...
Yeah. I mean,
No,
you have
I-
some habitat typologies that exist
Yeah.
in Europe, and
I
could be very interesting. I mean, when you're doing research to inform conservation, usually some of the basics are just to provide the habitat distribution within the.
So we were
area
in
of
contact
interest.
with the INBO lab that provided us with the emergent time data, and they provided us only with this as being habitat supralittoral to shallow sublittoral.
That's interesting, but it's still not habitat.
It's still not habitat, I agree. I worked with what I got. But
I understand. I'm not blaming you for that. I had the last question, but
The last one, right?
yeah, last one. Yeah, you say that there are some conservation actions that aim to maintain specific sediment characteristics of fauna. How
Yes.
did you do that?
How did they do that?
Yeah.
It's in the Sandbergen and Herrier
I didn't read those.
paper. They were the first one to recommend the actions in the Zwin Lagoon, and I visited the place even if I didn't go sampling, and what we can see is that so they limit the access of public every day to the area. There are forest guards that check that nobody is stepping on the protected zones, and I guess that they measure.
Is it enough to maintain sediment characteristics?
I don't think so. I think if
Because there's, I think you said the sediment deposition is a big issue.
yes,
Because of Lagos.
they have dig the channel 3 meters deeper in 2014.
Yeah.
So those are some of the restoration efforts that were done.
Okay, thank you very much.
Before we let you go, I have a comment and a question.
Yes.
I agree with Gauthier about the models would have been interesting to include multiple variables and just multiple regression, and you can have interaction among variables. You can have low order polynomials of some of those if you think the relationship is not linear. So that could have been interesting. And I'd like you to explain something. It's my understanding that a clustering algorithm is meant to cluster together things that are similar to one another, right? And how do you reconcile what we see about cluster 5 in your NMDS plot and the fact that all these these samples are clustered together?
I think so. The data I used for the cluster was only my macrobentic count, and I think that these were clustered together because these are the ones that had one individual, two individuals maybe, and this one had 300.
They should have low dissimilarity, and this
there's
plot
one individual of Arenicola marina, one individual of Scrobicularia. It's super.
I think the better is that this NMDS is not representing that very well because the stress is high and it's also a
Yes.
suboptimal representation.
It's still below 0.2, which is enough to have a pretty
Yeah, but
good
you probably would see a different picture of that in a metric, like a PCOA or something like that.
Yes.
That might be closer to what you get in the cluster.
I tried to have a PCA, but my data didn't meet the assumptions. I tried to transform them with a Hellinger transformation to have them meet at least normality, and it was still
You
not.
don't need normality, you need like at least some kind of symmetry, but that's fine.
Yeah,
It's a
I
technical
tried the
issue.
PCA, and as it was not conclusive, I just went with the
All right,
NMDS.
thank you very much.
You're
Elena.
welcome.
It's really open, right?
Alright, can I have your attention, everyone? We are live again, please, everyone. Thank you very much. So Mathieu, whenever you are ready, you have 15 minutes.
Hello everybody, my name is Matthew Legurin, and through the supervision of Carl van Collen, I'm here to show you my thesis, the Zwin Lagoon, how management reverberates throughout the trophic levels. So starting with salt marshes, they are similar neat environmental niches to mangroves, where they provide shelter and food as a nursery, so for juveniles, while also these juveniles are also a food source for larger prey items. Salt marshes are particularly impacted by the tidal forces more than mangroves, and they are important for humans in terms of they act as a barrier to floods and other coastal issues such as erosions. They are particularly vulnerable to climate change, and with the rise of human activity such as land reclamation, we are seeing worldwide a reduction in the amount of salt marshes. So we expect over time that there will be an increase in management actions for restoration of these salt marshes. So here we're looking at the Zwin Lagoon. It is located on the border between Netherlands and Belgium. It used to be the exit for Bruges, and now over time with land reclamation, sedimentation, it is no longer connected anymore, and instead it is just a lagoon, and it's an important place for bird biodiversity, and it was first recognized in 1952 by the king for that importance. So naturally, with there's a natural evolution of some marshes with sedimentation, and so even though it is a natural process to try and maintain bird diversity, there was a plan started in 2011 to restore the salt marshes through many different actions, with the one that I'm going to focus on today is the main channel, this one right here. It was, the width was increased and the depth was increased to increase the amount of water movement in and out. And so while this project is finished and it was deemed a success for birds, where one of them was to get the islands and as shown here, there is still further research needed to identify the other parts of the ecosystem, how they've been affected. So to do this, we're looking at two creeks, Creek 1 on the left and Creek 4 on the right, and we're looking at three levels. We're looking at the impact of the potential change in sediment, the potential change in microbenthos, and try to identify any changes in nekton habitat use. So these two creeks are located on each side of the main channel, and there is a change in elevation of these creeks. So we assume a change in inundation time compared to each other, and also with the digging of the main channel, we assume a reduction in inundation time, as now the water needs to rise up to higher levels in terms of relatively speaking before each of the creeks gets inundated. So for the sediment and macrobenthos data, along the creeks through randomized stratified sampling, we have 9 replicates for each of the years. For Creek 1, we have 2010, 2013, 2022, and 2023 data. Unfortunately, 2021 was sampled in the wrong creek, so it was sampled in Creek 2 just about, so it cannot be used. And Creek 4, we have 2013, 2021, and 2022 and 23 data. So for Necton, how the sampling is done is there is 3 mesh sizes with 3 nets. In the main channel, there is a 15 millimeter 5 net, and to try and have a more a better perception of the samples, there is a sub creek where a 5 millimeter net was put in, and so everything has to go through it, and a 1 millimeter subsample. They were put in at high tide, and they were left until the end of the until there was no more inundation. And then to try and relativize the data, we only use percentages and not actual abundance. So here's an example of the difference between low tide and high tide for one of the creeks. And so onto the data. Creek 1, we see a statistically significant increase in fine sediment. We're using mud content, which, as Elena pointed out, is a measure of sediment size, so below 63 micrometers. And for we also measure total organic matter, and for Creek 1, we see a statistically significant increase as well. However, Creek 4, even though there is a reduction in spread, there does not seem to be a statistically significant difference over time. So we're using this, onto macrobenthos. The 2 main variables we're looking at is the species richness and the total abundance. So following the same pattern for Creek 1, there's a very obvious, there's a statistically significant difference between pre-management 2014 and after management, so 2022 and 2023, for total of species richness and total abundance. Creek 4 does not seem to have any change over time. We also looked at the Shannon diversity to try and establish how the community changed over time. With post hoc test, we can see that 2010 and 2023 and 2022, 2010 and 2022 and 2010 and 2023 are statistically significantly different. However, 2013 is not statistically significantly different between the other 2, and I'll get into that a bit later. And since we're looking at Necton population, in the literature, Hedystay diversicolor is one of the main prey items, and so we looked at how that changed over time, and that showed an increase. For Creek 4, there was no change for either of these 2 over time. So to try and look deeper into the change of the actual communities, we looked at the percentages of each of the species. So here are the 3 main species, Apocylota marioni, Heteromastus filiformis, and Hedystay diversicolor. And there does not seem to be a specific trend for how between each year the percentages change. Onto the Necton. So we try to do MDS plots for each. So the closer the samples, the more similar they are. For 1 millimeter and 50 millimeter, we tried to identify some kind of trends. We did not seem to find any, so we're going to focus on 5 millimeter instead. The squares are June, the triangles are October, blue is Creek 1 and red is Creek 4. And what's one of the trends or one of the differences we have seen, and also very important, remember that each plot is a specific sample. There are no replicates for any of these. So there seems to be a difference between June and October, but it's not necessarily super explicit. And for Creek 1, there seems to be a change where the pre-management actions are closer together and the post-management actions are separated, but we're still trying to figure out exactly what that means, because when you look at the species that are found, it's also a majority of 4 species, where from this, trying to identify how potentially the trend over time has changed is quite difficult, and the only one that seemingly has appeared is for October. This is only Creek 1. The percentages of Bomasticus microps gobi in red seems to go up, while Carcinius meanus, brown crab, seems to decrease. So for the change in sediment, if we there are certain bases that the slower the water velocity, the lower the sediment that is able to be dropped off is. So if this is a not to scale representation, but the idea is there, where we expect that since Creek 1 has lower inundation time, as seen in the on site by 2 hours, we assume that now the sediment deposition is more focused around slack tide, which is where we'd expect to see the most amount of fine sediment being deposited. Fine sediment is also better at keeping the total organic matter, so we assume that the increase in fine sediment is the reason why there is an increase in total organic matter. When you look at the macrobenthos, here's an example of what they look like. The 3 main species, at least Hedystis diversicolor and Heteromassus philomoribis, have been tied to fine sediment, which is believed to be same with because of the increase in total organic matter, which they feed on for being detritivores. And Marioni does feed feeds on similar sources, but is not particularly associated with fine sediment, so it's a bit maybe surprising that they've increased. So Nekton, this is a part where it's a bit hard to try and identify how everything changes, or at least when we're trying to figure out the habitat use, because these are the 4 main species from the 5 millimeter, and they all can feed on polychaetes or on the or at least the 2 on the left explicitly can feed on polychaetes, while the 2 on the right can feed on are more omnivorous and can feed also on maybe some plant matter, and so there is an increase in food source for them. However, because of it's hard to quantify what would be the impact of larger predators, which have been seen in the 15 millimeter, but the variability is very strong, and it's hard to really show the impact onto them. We would have expected that a decrease of inundation time should lead to more of these species appearing, because they are more sheltered, as the predators have less, it's harder for them to get access to these areas. So to conclude, we have shown a potential link between how management has changed, has decreased inundation time, which should increase mud content, which in turn increases mud percentages, and how that is linked to polychaete abundance. One big limitation of this study is how, without any replicates for Nekton, based on how the sampling was done, it is hard to establish a trend for how that has been affected. And so if further research was done into that sort of area, even though it can no longer go back in time to pre-management, maybe a current research into how they move, how individual organisms on different organisms move around the lagoon for each of the creeks, where they spend more time, and how that is based on the inundation time for each of the areas. So yeah, and also thank you very much for listening. Thank you for Elena for her map. Thank you to my supervisor, who is currently on holiday, which after working with me and Elena for 6 months, he deserves.
But you got about a minute and a half to go, so that's fine. Thank you for the presentation. You want to go first, Mathieu?
I can go first. Thank you, Mathieu. That's a good thing you have the two together. That's kind of complementary. But I missed in your presentation statistics,
Yes.
and that's a big deal for me to say that. Obviously. Did you do any statistics?
Yes. For the it was we did Cruscal. I wanted to say it and I forgot. For the macrobenthos and sediment, we did Cruscal Wallace, and to try and identify the important species in the necton, we used Simper to try and figure out what.
Okay, it's important that you emphasize in your presentation the results that are significantly different and those that are not, because it really helps us to put a hierarchy in what you're telling us. You said something, and I would like to hear you about. You said that you calculated some Shannon indices to test for community composition changes. Do Shannon indices allow to test for community composition changes?
Okay, I think I said it's wrong. It was it's one of the variables to try and see, because one thing that we see is that for the species richness and total abundance, 2010 and 2013 are not very different, and then it goes up. But when we look at Shannon diversity, there's a slight increase.
Can you maybe go back to the slide position?
There's a difference between 2010, 2013, and 2013 and 2022, and we assume that while there is an increase in species richness and total abundance, the majority of the total abundance is actually an increase for the three main species. So the Shannon diversity is not as the difference is not as strong because it's not a equally distributed increase for all the species.
Okay, what affects Shannon diversity? Shannon diversity depends on two different things.
Yeah, species richness and total abundance.
Yeah, not total abundance.
The abundance for the individuals.
It's the same.
Oh,
it's species richness and evenness.
Oh, yeah.
Yeah, okay. What's the question? Yeah, can you go back now to the necton species? You only have 4 species.
For the main, the main ones, yes.
What do you mean by main?
Well,
this
is these are the main species.
What was in the others?
We had European sea bass juveniles. We had some anguilla juveniles. We had some a bit of amphipods and what's up in my head, and Mycedia.
Okay. Yeah, I don't know if it was a good idea to group them in other species, because actually you're focusing on the least informative species. Like the it's Pomatoschistus, not Pomatistus or whatever you say it.
Yes.
Pomatoschistus macrops is basically present in any single lagoon in huge numbers.
Yeah.
And if you're interested in the ecological functionality of the lagoon, it's probably might be a good idea to target species that are maybe less abundant.
Yeah,
that will be very sensitive to conservation actions, such as the European sea bass.
Yeah, it's so one of the things that's a bit hard is because there can be such a variation from June to October, or from one year to the next.
Yeah.
For Quick 4, we only have 2 years, and for 2023, from June there's about maybe I think 50 microbes,
Mhm.
and in October there's 3,000.
Yeah.
And that is a huge change, but we don't know if it's actually an important change or not, or if it's an issue with the sampling, or if it's an issue with the. And when we do the simpler analysis to see which are the species that affect the most the differences, these are the species that are that come up. So
Okay.
it's the perspective of how to do this analysis is something that is still trying to be figured out explicitly, and it's still not super happy with.
Okay. You talked a little bit about the relationship between these species and the benthos.
Mhm.
You say that they can all feed on benthos, which I agree.
Yeah.
But do you have an idea what they're actually feeding on?
I know that microbes and meanus have explicitly been found to feed on Ediste, while Rangron and Variance, they don't have, I'm still trying to figure out, I don't have an explicit species, and they feed more on plant and with dead animals.
Well, they can feed on pretty much anything.
Yeah.
And Kong can also feed on general flatfish, on Ediste as well.
Mhm.
They can feed on. Okay, and just another detail. We go back to the photos of your species. Makes my eyes bleed. It's Carcinus minus, not Carcineus minus. That's important. Thank you, Mathieu. Thank you.
Hello. Oh, well, let me start with clarifying something I may misunderstood. So the opening of the channel was 2014?
14.
So you have one sample before and three after?
For Creek 1, we have 2 samples before and 2 samples afterwards. For Creek 4, we have
One
one
and
before
three.
and 3 after.
So the purpose of your work was to see the influence of opening the channel on the diversity of these ecosystems.
Kinda, there's a lot of assumptions in terms of like we assume that they are being affected, and so with the current because the sampling has already been done and the sampling was not done in specifically this for this project. Yes, that's what we're trying to see, but
So you're assuming that possible changes over time were not
Yes.
relevant for this analysis?
we didn't take into account because like one thing that would have been nice would be if we had any hydrological data to see if the actual water velocity did change over time, but we do not have this data, and so we assume that the change in sediment is based on the inundation time, which is based on measurement, but it's assumptions, and so
Okay. And I understand you have one sample on each creek, each season, each year?
yes,
Okay, so no issues, we're not having replicates?
lots of issues. But it's those one of the limitations of trying to look at ways to analyze the impact of a project afterwards with how they decided to implement the sampling for them to see how it changes over time. Where if I was doing this from the start, I would have not done this kind of sampling for the Necton because without any replicates, we cannot identify a trend, either positive, negative, the same, and so yes, there's a lot of issues with having the replicates, and this is why.
Whatever the purpose of the project was, no
Mhm.
replicates kind
Yes.
of makes it
Yeah.
strange. All right, then you said something that opening a channel reduced the inundation time of the smaller creek.
Mhm.
Why?
So with the widening and especially the deepening is because that based on the tidal times, it requires the water to go up higher for it to inundate onto the creeks, and so there has been a in situ because of they measure how long they put in the nets. There is a reduction of from 6 to 4 hours, and so like I said, there is an assumption that because of the deepening that it takes longer for the water to go up than inundate the creeks, but we don't have a
Would it be
direct.
the speed of the water
yes.
that is lower because you
Yes.
have a much, it's
But
narrow,
we assume so. We don't have any direct.
you don't have measurements of
We have a measurement of inundation time between 2013 and 2023, which has been went from 6 hours to 4 hours, and so we assume that is because of management.
So my last question is the result of the previous ones, not this one about the channel. Do you feel confident to do recommendations for management of these based on your data?
I'm for sediment and macrobenthos, I'm more confident because it kind of fits in line with what we might expect with the literature. For Necton, I'm not competent to say anything because I have nothing to explicitly state out in terms of the management for any of the species.
Okay, thanks.
All right. I have a few questions, and I'm sure we can go back to Gauthier afterwards because I'm sure he has more. Just a common misconception about the results of the Simpro analysis is that it identifies species that are characteristic of a given group.
No.
But that's not what they do. They
It's the one
point
that
out
shows the biggest difference between.
yeah, so you could have a species that's everywhere in all your samples, but
And
if in a group there's just a little bit and in the other one a lot, that's gonna come out.
And because we're doing
Right.
percentages, sometimes we have 0 and then it goes to maybe only up to 3 organisms, but because there were no other organisms, it shows a massive increase.
So,
And
yeah,
because
so
we,
you're aware of that,
we
yeah.
actually have no replicates,
Okay.
it's,
Okay.
yeah, that's the issue of only having one.
Can you go back to the sampling, because I just because I'm not completely clear in my head on like, if the 2 creeks, what you have is in each ear, so.
This is not for all the years. This is for 1 year specifically.
Okay, but for let's say you have, or maybe you can show me on the NMDS plus because they're all there, right? Your samples.
But for MDS, it's for the Necton. This is the sediment samples.
Okay, but then I'm interested in Necton, which is your main concern. So you have the 2 creeks, and then you have 4 ears in each, but they're not always the same, right?
Yes.
And are some of them the same?
So 2013 is in both, 2022 is in both, and 2023. But there was
Okay.
no sampling for 2010.
But you could say, yeah, but you could still say, you could group the 2 together. Because what I'm wondering is, you could have maybe at least tried to test ear or creek and to see in your community if there's a difference, right? Because you did it for the alpha diversity, couldn't you have done it for the, because you have, if you consider your creeks as replicates of an ear, couldn't you not, you only have 2, I agree, but couldn't you have tested the ear factor? And vice versa?
Yes. It didn't really come in. I didn't really think of it yet. So, because for us, the important part was not how the change in macrobenthos and sediment was not necessarily the most important part. We're trying to focus on the final one because, like Elena. Her work is a recreation of other papers from 2014, which have looked at the relationship for sediment and macrobenthos. So that's something that even in the area has already been more explicitly elaborated on.
Okay.
And so the original idea was not to focus too much on these 2 and try and do the final part, which is not something that's been done in the.
Okay. But just saying on the topic of the replication, I'm not sure the creeks are replicates of the ear because you've showed some differences, so you could have issues with that. Just, I don't know if it's worth it for you, but you can look at the method that actually was developed for monitoring, where we often don't have replication, either in space or time, and it's a clever way of still being able to test the interaction of, for which you need more than just 1 replicate, between space and time, in your case, creek and ear, and it's a method developed by Michael de Caceres. It's from 2003 or 2001. Look, testing space-time interaction with no replication, and you can actually do it. It's a very clever way of looking at it.
Okay.
Yeah, good say.
No, maybe just a small question. You had lots of dead pomatoschistes. Did you try to open them, or to open their guts to see what was in there?
No. This is not
How come that? That would be a good idea. This is what the title of the topic is how conservation measures reverberate through the trophic levels. That would be a cheap and easy way to understand.
It was not in it was not in the plan of how to do it. I don't know how well the actual insides keep, because most of these samples are years old in formaldehyde. I don't know.
The formalin, they can be hundreds of years old, and still.
Yeah, we know we didn't. It was not something we looked at.
Okay.
All right. Thank you very much, Peter. We will move on to the maybe most or the least desirable spot in this symposium, depending on the way you see it.
Final stretch.
You're the final one, you're the last.
Just a little longer.
Yes.
Thank you.
So we welcome Emily.
There was a very nice picture here, but apparently not.
And Emily, whenever you are ready, you can start in 15 minutes.
Thank you. Operating under false assumptions is something we've likely all done before. Unfortunately, when it happens in conservation, it can have dire consequences. It can cause a sudden population collapse or even a local extinction. And I worry that is what's happening with the black-spotted smooth-hand sharks in the southern Atlantic Sea. This mesopredator is incredibly valuable to the coastal waters and continental shelves of the Mediterranean and the central eastern Atlantic. Additionally, it's economically very valuable, as it is known to be targeted by fishermen across Mediterranean cultures. However, it is listed as vulnerable under the IUCN, with a population decline recorded at 49% in the last 3 generations, and it continues to decline. Yet almost nothing is known about this species population, especially not in the southern Adriatic Sea, and that information that is available is not always entirely correct. One of the reasons for this is taxonomical issues, as this species, the Musculus punctulatus, shares a lot of morphological similarities with the Musculus musculus, its sister species. So how do we differentiate between the two? The most important feature is the snout, which is rounder in the Musculus punctulatus. You can ensure this by looking at the internasal distance and the nasal length. Additionally, the presence of black spots and frayed edges of the caudal and dorsal fins can indicate the presence of black-spotted smooth-hand shark, but their absence does not mean it's a common smooth-hand shark. This, plus lack of funding in certain countries in the southern Adriatic, can cause very little information to be available. Therefore, I, Emily de Roze, along with my supervisor, Dr. André Gage with Shark Lab Adria, decided to provide the first current population status of the Musculus punctulatus in the southern Adriatic Sea, as well as assess several of the anthropogenic pressures, as we believe that there is a resident and healthy population that is suffering. You can see that this is a broad research topic, and therefore my methodology will also be rather broad and multidisciplinary. We start by sampling in the field, this time not in Zvin, but we go to Albania in Vlore at Shreveport Harbor. This is the main harbor of Vlore, where we collected black-spotted smooth-hand sharks from commercial and artisanal trawlers that would fish at depths between 50 and 600 meters. Deceased individuals we would bring back to the lab, while the live ones we would attempt to rehabilitate and release. It is incredibly important to note that no individuals were caught, killed, or injured for the purpose of this study. Additional fieldwork included habitat analysis, where we would take smaller vessels to the Vjosa Delta, Karaburun Peninsula, Southern Island, and Flora Bay, with a Triton open ROV. This would survey, and then the footage would later be analyzed for the bottom type, the potential prey species, predator species, or signs of the elasmo branch themselves. The final part of the field work was collecting local ecological knowledge to supplement the knowledge that we did not have. In conversations with the fishermen, we would be able to assess the catch rates and how that had fluctuated over time, between seasons and in locality. And doing questionnaires at fish markets would give us the market value of the balloon, or the smooth-hand sharks, and changes in availability that they would have noted as well. Back at the lab, we would make sure to meticulously identify our individuals and then measure them. For the first time for this species, we calculated biometric parameters that would allow us to know the overall well-being of our samples. The condition factor compares the total weight and length to give the overall condition, while the hepatosomatic index was used to quantify the lipid storage and the energy storage in our specimens, and the gonadosomatic index used the gonad weights to give an approximation for the maturity and the reproductive stage. We also did a stomach content analysis to be able to compare the diet composition against the population of the wider Mediterranean by isolating both functional parts of the stomach and identifying the contents to the nearest taxonomical level on a visual basis with a stereo microscope. The largest part of our work in the lab was spent doing autopsies as part of a health assessment. We would make sure to really inspect every individual for signs of physical trauma, barotrauma, alterations in the major viscera and the skeletal-muscular system, or pathologies. We also did parasitological examinations to identify any endo or ectoparasites, and got the wonderful opportunity to do an MRI scan of an individual as well, which could give us a view of the internal anatomy so that it would not be disrupted by dissections, and give us a clearer view of maybe liquid buildup that could sign a pathology or breaks in the cartilage. This could help us with post-capture survival assessments as well. And part of our health assessment was the melanomacrophage analysis. Melanomacrophages are part of the non-specific immune system in elasmobranches, branches, and they're used as a bioindicator for water quality in fishes. This is now also being evaluated for their use in elasmobranches, branches, which we wanted to participate in. So we sample delivered of 15 sharks, had histological slides made, and then identified the melanomacrophages so we could measure them, and this would give us an indication of their response to the water quality. The final part of our work was trace element analysis, which was also the first time this was done for our species. We sampled the liver, the kidney, the brain, and the muscle of 13 individuals and dried these samples and sent them off to Italy, where Dr. Annabaldi and her team kindly helped us at the Université Polytechnique del Marche with atomic absorption spectrometry for the selenium and the mercury content, and atomic emission spectrometry was used where we were specifically interested in the lead, the arsenic, the chromium, and the cadmium concentrations, as these can be quite dangerous for human consumers as well as potentially the elasmobranch. So, what can we say? We'll start with our samples. We collected 22 free-living individuals. 12 of these were adults, 8 of these were juveniles, and 2 were neonates with an umbilical scar. We also had a litter of embryos that still were attached to the yolk sac, and 3 litters of late-term fetuses that had formed a placental connection. The free-living individuals could give us an idea of abundance. You see, previous studies had found 5 individuals caught over the course of 2 years in the entire Albanian fleet. Additionally, 151 individuals were identified in the method survey, which took 2 years and wanted to assess the demersal stock of the Mediterranean. We found 22 free-living individuals from 1 port in Albania over 2 years, which appeared to be a rather average catch rate according to the fishermen. And our fish market survey showed that usually 1 large juvenile or adult individual was available for sale per week in most markets. If we extrapolate this data based on the registered Albanian fleet of trawlers, we find closer to 30,000 individuals being caught every 2 years in Albania, compared to 5. So this shows us that there has been a significant underestimation of abundance of black-spotted smoothhound sharks in the southern Adriatic. They're likely much more prevalent there, and the fact we found no common smoothhound sharks could suggest that they're the dominant smoothhound shark in the area. It also notes that the fishing pressure is likely severely underestimated, as those numbers were also based on these studies. What can we say about the local ecological knowledge? This provided us with information that very small, likely neonates, and large gravid females were caught in Karaboeren Peninsula, Sazan Island, Vlora Bay, and the Vjosa Delta in the late spring months. This means that these areas could be potentially nursery areas off the coast of Albania. Their presence, as well as recently mated males and all other life stages, means that this is likely a resident population in these waters. And luckily for us, this population also appears to be in overall good condition. An average condition factor of 0.37 and a hepatosomatic index of 6.07, yes, is within the range that we would expect for this species. I was able to compare the condition factor against those I calculated for the black-spotted smoothhound sharks in the northern Adriatic and the Tunisian waters, and found ours to be slightly elevated. The hepatosomatic index approximated those of the common smoothhound sharks in the wider Mediterranean. Unfortunately, however, the gonadosomatic index we did not use, as the gonads were often too decomposed to use for proper measurements, which was unfortunate. The ranges for the other values did also fall within line of what we would expect. The diet composition showed a large overlap with other populations in the Mediterranean, which we would expect as well, with a high preference for crustacea and an ontogenetic shift to include more cephalopods and teleosts as the individuals were able to catch the faster swimming prey. We did note, however, that we had a lower diversity with a significantly higher preference for crustacea, especially for the Squila mantis. So this could be due to a range of reasons, maybe a lower interspecies competition with the common smoothhand shark, the different bottom habitats, the shorter time frame of our study being 3 months, or maybe a fourth one, or the combination of all actually. The health assessment also showed us that our population appeared to be overall quite healthy. We found 2 major alterations: an acute superactive otitis media with concurrent inflammation of the endolymphatic ducts, or an inner ear infection, and a runt amongst a litter of normally developed late-term fetuses that was likely stillborn. Other than that, we found no real signs of major alterations of the viscera, breaks in the cartilage, signs of physical trauma, or other pathologies. This seems to mean that these individuals are very resistant to being caught on a trawler and handled on the deck of the ship. This, plus the fact that the individuals we attempted to release seem to swim away independently, rather reliably, means that they could have a very high post-capture survival rate. No physiological tests on stress have yet been done though, so they might still die or suffer from the stress at a later date. Still, it is hopeful. From the parasitological examination, we found 3 taxa of endoparasites in the gastrointestinal tract, which were normally found in the Musculus genus. And the melanomacrophage analysis was simply baseline data that showed a closer approximation to the demersal elasmo branches in the southern Adriatic, rather than the pelagic species found in the American coast. So, what is our population suffering under? I already hinted at the anthropogenic pressure that was the most problematic is the fishing pressure, as it was severely underestimated. Both fishermen and fish market employees noted that there has been a decline in stock and availability, though the exact number cannot be deduced based on this. One fish market employee was very insistent that there had been a 50% decline in bloom availability over the last 20 years, which is quite alarming. And this is only aggravated by the conservation measures in place. Albania is required to protect all smoothhand sharks and regulate their exploitation under the Barcelona Convention. Yet the minimum landing size that they have in place is 30 centimeters. Based on our study and confirmed by literature, at size at birth, they are 24.6 to 36 centimeters. There you go. And they do not reach maturity until at least 70 centimeters, meaning every individual caught can and will be landed, meaning this minimum landing size is essentially obsolete. We also looked at the trace elements and found no immediate correlation between pathologies or maybe biometric parameters that showed any difference. Therefore, we believe that it did not affect the elasmo branches as severely, which is confirmed by literature, but much more research is needed. The top 4 elements we found was iron, zinc, aluminium, and arsenic, which is regularly found in higher concentrations in aluminium, with iron and zinc being essential trace elements, and arsenic being especially dominant in the demersal species that like to feed on crustacea. We were able to compare our concentrations against the Musculus of the south of Italy and as well other elasmo branches of the Mediterranean. And most interestingly there was the nickel concentration, which appeared to be significantly lower in our population compared to the Musculus. In the muscles it was about 20 times lower, while in the brain it was up to 90 times lower. But if you look at the values of the other elasmo branches in the Mediterranean, ours is actually slightly higher, so that was quite interesting. The mercury and the copper concentrations, however, were also slightly lower than those in the Musculus. Most concerningly, though, about the trace elements, what we found was for human consumers, as the muscle concentrations showed were higher than the maximum regulatory limits in mercury, arsenic, and chromium, especially in arsenic, where all of them exceeded the maximum limit, and occasionally 18-fold, meaning the meat is highly toxic, cancerous, and can cause stillbirths and developmental disorders. So that's quite a big problem. The habitat analysis, unfortunately, was not able to be done to its full extent due to weather complications, so we only surveyed a small part of Flora Bay, which did show some signs that it is perfect habitat for the Musculus punctuatus. So, in conclusion, we have a highly abundant population of black-spotted smoothhound sharks in the southern Adriatic Sea that was severely underestimated before. It is likely in very good condition and quite healthy, though it does seem to be suffering severely under the anthropogenic fishing pressure that is aggravated by inefficient conservation measures. Trace element analysis might be cause for concern for the species, but is more important for the human consumer. It is incredibly important that we do more research on this species to ensure that this beautiful animal does not disappear and effective conservation measures can be put in place. Before I drop you guys off at the Q&A session, I just want to note that we are working on publishing some of these results, as well as results about other species that we got to work with in Southern Adriatic. Thank you so much for your attention, and I'll
Yes.
Thank you for this, for your work, for the presentation, very interesting work, and very professionally delivered presentation within like 15 seconds of the allotted time. So.
I could have taken a couple extra reps.
Yeah, exactly, but not many. So I don't know who wants to go first, Margarita or Gauthier.
Hello, very, very well presented.
Thank you very much.
You mentioned that you sampled in the ports. What was the gear they used?
So the trawling nets were very standard bottom trawlers, and then they had long line fishing nets as well, and there was some variation between the general equipment when it comes to mesh size.
So trawling and
long
line.
yes. But most of the samples did come from the bottom trawlers, as these were much more, let's say, efficient in catching the investment batches.
Did you ask them if they throw away a lot of individuals? What is the target species of the trawling?
So the trawling is very unselective, as many people know. The black-spotted smoothhound shark is one of the targeted individuals, but they essentially try and catch everything that they can of the demersal stock. And they have an extreme amount of bycatch, especially, for example, cat sharks, both the small spotted and the blackmouth cat sharks. And these are discarded quite regularly. One of our fellow colleagues did her thesis on one of these bicot species, actually. So it is very unselective, and they try and sell most delios that they can catch, as well as certain, like the shrimp are, for example, very popular as well.
So I guess that shrimp would be the target species, and it's the most valuable.
This is the one that they're most excited about having, but they target everything that they can.
And the other question is, you looked at 22 adults,
yes,
and they were all from the harbors in Albania, collected for what period of time?
So that was 22 free-living individuals, including adults, and they were sampled over the course of 3 months. So from, let's say, mid-January essentially to mid-April.
Okay.
So also during the months, this is why we had the later fetuses as well.
Because the condition indexes you showed are cyclic along the year, so I'm glad you didn't use the autosomatic index, but even the other two are cyclic.
They are.
So looking at 3 months probably doesn't tell you much about the condition of the individuals if you don't have a full cycle for comparison.
It does, of course, yeah, much more research is needed, you're correct, especially like the hepatosomatic index can vary greatly based on the reproductive stage of the females. So it is necessary to like expand it. This was a preliminary study to see, we were mainly hoping not to see a drastically low number. So we just wanted to bring forth the first numbers so that this maybe can be compared at later times as well.
Another question I had is the distribution of the species.
Yes.
Where does it distribute in the Mediterranean?
So it is across the entire Mediterranean in waters ranging from about 50 to originally 300 meters, though we did find one at 350 meters. So this might be, just one individual that went deeper, or maybe a sign of something else. Except the Black Sea, they're not present there, and then they're found at the north coast of Africa as well.
So you have a species that has a wide distribution that very likely has exchanges because they're highly mobile along all this area, and you're focusing on a very small area. You're confident to say that the species is in a good status in terms of their condition?
So I feel, let's say, confident about this specific sampled population. Of course, there might be a big variety based on where we're sampling. The anthropogenic pressures might be very different, As we saw with the musculus, which the common swimfan shark in the south of Italy, the trace elements, they already showed big variance, and these are two very similar species in a very similar area. So of course there could be a big difference. The interesting spot about where we sampled is that it's right at the mouth where the Adriatic meets the Mediterranean, so we do have a lot of influx there. So that could, a lot of individuals might be passing through there, but it's likely also a population that seems to be quite resident. So they're less likely maybe to move to maybe North Adriatic or anything like that. But I cannot confirm that, of course.
One last question. You mentioned you studied macrophage aggregates, or you called them something else, but
Melanomacrophages.
yeah. You didn't show the results.
Yes, I did kind of skip over these a little bit, not because it's not interesting, but time limit, and it's simply baseline data. So melanomacrophages in elasmobranchs branches are a little bit more complex than entheliostes, as they are very much more based on reproductive stage, life stage, sex, and everything else. So the goal of these studies, all of these, including ours, is essentially to start building a baseline. As elasmobranchs branches are very long-lived, they could be very interesting bioindicator to see, the pollution of the water quality and how things are changing.
In principle, they're not excreted; they stay in the.
They're part of the immune system, yes.
And they are related with physiological age, so
yes.
You have to relate them with at least the size. I'm not assuming you're going to, because as they're older, they accumulate a lot more of this.
So it's the melanomacrophages themselves. They are part of the immune system, so they are not a pollutant themselves. They're responding to the pollutants in the water. So the more that are there is because there's more pollution likely, or their size is because they've activated and they've started doing their job essentially in the body. So it's just essentially histology that you're doing at that point. But yes, it is very dependent on life stage and reproductive stage. So much more needs to be done, and it's already being noticed. Like there's a very big difference based on the animal. Like the difference in metabolism, the life stages, like the pelagic species are clearly very different than the demersal species. So it's the very start and just baseline data that's been provided.
All right, thank you.
No problem.
Thank you, Margarita. Luce?
Thank you for this dynamic presentation. I was wondering about the fishing pressure. Do you know where the fishermen are fishing?
Yes, so the map that I showed is the general area that they fish. There is, of course, a little bit of variation, and depending on which trawler and longline, they might go further out as well. But these are the main areas that would catch the black spotted swordfish.
They basically remain in the bay.
So they remain in the bay, and then they tend to go around these areas as well. So it's not always very far. Many of the fishermen, they will do one-day tours. There's also occasional 4-day trolling tours, but these areas are actually seeming to be very abundant in a lot of organisms, so they tend to accumulate in these areas as well.
Okay, and do you know what is the depth distribution range of your species?
I didn't mention it in the presentation. Its original depth limitation is about 50 to 300 meters, but as I mentioned before, we did find one at 350 meters. The musculus, the common swimfan shark, is known to come deeper, so this could be a sign of a taxonomical, like morphological identification, causing some difficulties in the literature that has existed. So potentially they could be going deeper than their.
Do the fishermen fish that deep?
They do. They trawl deeper as well. They catch deep sea species quite regularly.
Okay. How did you extrapolate the catches when you took for glory to the entire companion fleet?
So this is a very essentially basic estimation. No models or unfortunately statistics was used here. We simply found the registered trawler fleet that they had. I believe it's 186 or something, and then based on the numbers that we had from our trawlers and trawlers that we were able to sample, we simply took these and extrapolated them. As based on the local ecological knowledge, this seems to be a very normal amount that they would catch throughout the year.
Okay, but do they catch the species throughout Albania?
Yes, it is also caught across the Atlantic. It's regularly found, well, I would say it's regularly found in the literature, but that's not entirely true. But in speaking with fishermen in other countries, you do know that they're often on the fish markets for sale.
Another question, maybe my last question about the hepatosomatic index.
Yes.
How do you interpret that?
So with the hepatosomatic index, this was mainly I compared it to other. I compared it to other literature. It's the higher the value, the larger the liver is the very basic concept of it.
Yeah.
And therefore, if we were to find very low values, we would be able to see that maybe there's a diet deficiency or a lot of the energy that has been used for maybe pathology or any kind of other issue that the animal needs energy to stabilize itself or maintain itself. So this was just to get an overview to see if we noticed any big changes with it.
Is the hepatic way related to metal contamination or anything else?
No, we found no link between that. What we did find, I did mention because time limits, is significantly more metals in the liver and the kidney, but this was expected because they participate in detox and excretion, so they had always much higher levels, which again, if you look at the arsenic, is concerning, as it was much higher there as well.
Okay. Okay, thank you.
Thank you very much.
You have more questions, Margarita?
No, I don't.
I won't ask you any questions, so thank you very much. Oh, there's a question from the room. Yes, please.
In my mind, sharks needed to be gutted really quickly to avoid any ammonia spreading into the overall system. So here, the sharks were pulled onto the fishing market.
No, not all. Most of the time, the sharks were gutted on the ship. Actually, some of my samples were gutted. I think we had one that the fishermen had already prepared and eaten on board, and they provided us with the organs and the head and everything else. So these here are both gutted individuals, and then here we see how it's most often sold. So it's completely prepared. Because many of the Albanians, there's a difference based on which background they have. They're actually not super, it's not considered a high value meat. They don't always know how to prepare it. It's more often sold to restaurants for soup in certain areas. So yeah, they prepare it quite quickly. Well,
Thank you. So congratulations to our last three presenters in this session, to all of you, of course, for completing this exercise.